Yeah, it's live for me.
Okay, great. It seems like we're live now, so we can get started. Okay, I think yeah, let's get started. Welcome everyone. I guess all over the world. We're having our Safari live seminar today with Damla Senol, who is a staff software engineer in the hardware acceleration division of Bionano Genomics company. And she's going to talk about actually what she did during her PhD thesis at Carnegie Mellon University as part of the Safari research group, co-advised by myself and Sogata Goes. And Damla was at Carnegie Mellon for about 6 years, doing essentially working on these topics and some other topics in memory systems and DRAM. Before that, she graduated from Bill Kent University, did her bachelor's degree over there. And during her PhD, she interned at Intel Labs, and she decided to very recently take a position after her PhD at Bionano Genomics, hopefully continuing some of the work that she has done over her PhD and getting it into real products, let's say. So we will hopefully hear more from her research and product experience going into the future. And she finished her PhD essentially a couple of months ago, in August, right? Technically, let's say August 2021. And she was my last student at Carnegie Mellon University. So Damla is going to talk about accelerating genome sequence analysis via efficient hardware algorithm co-design. This is a quite exciting topical area. How do we actually enable genome analysis to be much faster, much more efficient? And this requires hardware algorithm co-design. And she's going to take us through the journey she has gone through during her PhD, including analysis of the bottlenecks of some important workloads in genome analysis for long reads, for example, nanopore sequencing, and then some designs that she has developed to actually make genome sequence analysis much faster, especially focusing on approximate string matching, and then some other things that she has continued to do and that have led to significant performance and efficiency improvements. But I'm not going to tell you all of that, so I will let Damla talk about it. Damla, the floor is yours. Thanks for doing this.
Thank you so much, Onur, for the introduction. And it was an honor to be part of the Safari team. And right now, it's an honor to be here. So thank you so much for joining my talk. Onur did the introduction, so I will skip that. But just to summarize again, today I will be discussing some of the work I did during my PhD on accelerating genome sequence analysis via efficient hardware algorithm co-design. But let's start with some background, and let's start with genome sequencing. So genome sequencing enables us to determine the DNA sequence of an organism, and it plays a pivotal role in areas such as personalized medicine, outbreak tracing like COVID-19, and the understanding of evolution. However, there is no sequencing machine that takes the long, complete DNA as the input and gives the complete sequence as the output. Instead, the sequencing machines extract small randomized fragments, like chopped fragments all over the full DNA sequence of the original DNA sequence. So when we look at the process, as you can imagine, it starts with the sample collection. It can be blood, saliva, some bone tissue, something like that. And the goal is to obtain the large DNA molecule out of it. But it goes through a wet lab preparation process. And in that process, we divide the full DNA molecule into chopped DNA fragments. And then those chopped DNA fragments are the inputs to the sequencing device or any instrument that is responsible for gathering that genomic information out of it. And in this case, it's a sequencing. And after sequencing step, we get the sequence reads. What I mean is the strings, which are composed of ACGTs. But as I said, since we chop them and they are mixed, they are kind of randomized in terms of location, and they are they represent different portions of the original full DNA sequence, and we lost the location information actually. So in order to gather the information that we are interested in, the meaningful information out of these sequence trees, we have to perform genome sequence analysis. And this is where the genome sequence analysis term comes from.
So Damla,
so there are, yes.
I have a question, the previous slide.
Of course, go ahead.
Yeah, about these pieces coming from the sequencing machine, how large, how long they can be currently?
So that's a really good question. I think nanopore technology, which is one of the the newer technologies, they are looking at millions of they call them ultra long DNA molecules. So we are talking about millions these days, but it is not the average. We cannot say it average because other sequencing technologies, which I will show in my next slide, like Illumina, for example, they are talking maximum of 500 characters or 500 bases, we call them. So it's a very, very large range. But as I said, one of the the leading newer technologies, nanopore sequencing technology, are shooting at around millions these days. But for example, PacBio, which is another kind of newer, again, long read technology, their average is around 30 kilobases. So as I said, the range is large, and we cannot say that millions is the average, but it's definitely getting improved for sure.
So why we should deal with the short pieces? Isn't it long pieces much better, easier maybe to deal with?
Because they are cheaper. So the technology itself, of course, comes with its own cost. And right now, the long read sequencing technologies cannot beat the cheap cost of short read technology like Illumina, for example. So there's a cost relation there as well. Yes, maybe long read technologies are producing more longer reads, but they are not cost efficient. So that's one reason. Yeah.
And how do you envision the future of these sequencing technologies?
That's a good question. I'm kind of a bit biased because of my current company as well. But when we talk about sequencing itself, for example, one trend might be in the, so I don't believe that short reads technologies will be gone. And as most of us who are from this field know, that Illumina is a very huge company, which is the leading technology in short reads. So I think longs and shorts will continue going together. But also we know that and why we know that, another indication is long read companies are interested in combining some other short read companies in their platform as well, so they can provide both. So it's another indication that none of these, only one of these types will be leading in the future. But my belief is none of these will be gone. But we know that long reads are companies are decreasing their cost as well. So it's a bit a tricky, hard question to answer, but I don't believe that either of these, so long reads cannot be gone for sure, because they are very, very promising and providing a lot of benefits on top of short reads. But cost is a significant issue, of course. So I don't know at one point whether they will be able to beat in terms of cost, the short read technologies, actually.
That's interesting. Yeah, I agree with you. Yeah, thanks.
Yeah, of course. But it was a really good question, I think. So we cannot know the future, of course, but we are just throwing some ideas, what we believe and what we see, the trend itself, mostly from the company side, because they are leading the domain. So yeah, we as researchers kind of providing support to them in terms of us, for example, in the computation side. So we will see. It's interesting. Yeah. Thank you. Is there any other question before I move on? Okay. So I mentioned these technologies while answering Mohammed's question, but as I said, there are multiple sequencing technologies. One of them is Oxford Nanopore, which is a newer company. They're not new anymore, but newer relative to Illumina, and they're producing ultra long reads, actually. There is PacBio, which is another long read sequencing technology. And we also have Illumina, which is the short read sequencing technology, and we have more than these, more like smaller ones as well. And what is important in terms of computation here is, so they are producing different types of reads, short reads, long reads. And why we care them? Because in short reads, yes, we are dealing with just a few hundred base pairs, but they are extremely accurate. So the error rate is very, very small, and these percentages are for the read length. So among the read length, is around 0.1% error rate we are dealing with in the short read scenario. On the other hand, in long reads, we are dealing with thousands of millions of base pairs. It is great, but the error rate that we are dealing with is much higher compared to short reads, which is around 5 to 10%. This number is getting decreased, but still we are dealing with 5 to 10% these days. And again, this is out of the read length we are dealing with. So there is not one single type of read that's winning in both of these metrics. And when you look at the trend in terms of sequencing, so the sequencing device is becoming cheaper, more accessible, which means that more people have access to these devices, more data is being generated. So the amount of genomics data that's generated getting much and much higher. And these are from Oxford Nanopore and Illumina. Let's look at the Illumina. This is from 2014, so 7 years ago. They were annually generating 18,000 of full human genomes, like sequencing. So it's a huge amount of number. And as you can imagine, this number is getting higher and higher. And however, we know that the compute power doesn't match up with this trend. When you look at Moore's law, for example, the green line shows the cost of sequencing a single human genome. On the other hand, the white line shows the Moore's law. So modern sequencing machines generate read fragments at an exponentially higher rate than prior sequencing technologies, with their growth far outpacing the growth in computational power in recent years, which means that computation is becoming the bottleneck. So you can generate a lot of genomics data at a very fast pace, but if you cannot keep up the computation size, which is genome sequence analysis or genomic data analysis, whatever you call it, if you cannot keep up with that, then it's meaningless, because we are gathering the information after the computation step. Yes, generating a lot of genomics data is very, very beneficial. It's useful, but we have to keep up with the rate of it. With data generation and data analysis rates should match. So in this case, computation is becoming the bottleneck, so that we cannot exploit full of the benefits that the sequencing technologies are providing to us. So the problem here is, so rapid genome sequence analysis is currently bottlenecked by the computational power and memory bandwidth limitations of existing systems, since many of the steps in genome sequence analysis must process a large amount of data, and the amount of this data is getting higher at each day, let's say. So to this end, our goal is to accelerate genome sequence analysis with efficient hardware algorithm co-design. So we need to understand first where the current tools and algorithms do not perform well, so that we can develop better tools. And also we have to understand the limitations of existing hardware systems when running these tools and algorithms in order to design efficient accelerators, like customized accelerators. So our approach is to first analyze the multiple steps and the associated tools in the genome sequence analysis pipeline, then expose the trade-offs between different metrics like accuracy, performance, memory usage, and scalability, and also reveal the bottlenecks for these metrics. And finally, co-design fast and efficient algorithms along with scalable and energy efficient customized hardware accelerators for the key bottleneck steps of the pipeline.
So Damla, I have a little bit of debate about the goal here. So why would someone worry about having his genome analyzed really quickly, or what is the need for accelerating that process? For example, if the clinic needs 5 hours to give you the results, I can wait that long. If it is 7 hours, also it's fine with me. Or if it is a long day, that is also fine with most of us. Why we should worry about accelerating this step?
Because there are some diseases, especially in the baby phases, like newborns, this is just an example. There are, I'm pretty sure, more and more about these kind of examples, which is extremely time critical. And you cannot afford hours in those scenarios. You don't want to. That's one reason. The other reason is scalability. So yes, you might afford 5 hours dedicated or a day dedicated to you, but if you want to scale the amount of genome sequence analysis, let's say personalized medicine, which involves like analyzing the genomic data for multiple people. So if you want to scale, then you need to be able to decrease the cost that you are dedicating for one sample. So yes, you might have wait for it, but if we want to have billions, thousands, whatever the scale will be, then the time will be extremely critical. This is the second reason. So, but of course, when we think about the first scenario, it sounds much more critical because you have to be able to answer in a very short time so that you can save that person, that baby, whatever. So if you want to, you have to be able to gather that information on time. And there are some scenarios which you cannot afford hours. So that's why it's super critical. And as I said, it's becoming more accessible, meaning the scale will be much higher than today in the upcoming years, let's say. So if you want to be able to afford that, then you have to accelerate. And cost, of course, is another metric as well. So cost in terms of not just money, but everything, like time, everything. So you have to decrease that as well.
I like that. Yeah.
Oh. Good. Any other question? Thanks, Mohammed. Okay, I'm continuing then. All right. I think, all right. So I stated the problem, and in aligned with that, my research statement is as follows. So genome sequence analysis can be accelerated by co-designing fast and efficient algorithms, along with scalable and energy efficient customized hardware accelerators for the key bottleneck steps of the pipeline. And towards this end, we propose 4 major works as part of my PhD thesis or PhD work. So the first one is where we published in Briefings in Bioinformatics 3 years ago. So in this work, we comprehensively analyzed the tools in genome assembly pipeline for long reads in multiple dimensions. We uncovered bottlenecks and trade-offs that different combinations of tools and also different underlying systems lead to. And we showed that we need high performance, memory efficient, low power, and also scalable designs for genome sequence analysis in order to exploit the advantages that genome sequencing provides. And then in our second work, we proposed GenAsm, which we published in Micro last year. And we proposed GenAsm, which is an acceleration framework that builds upon bit vector-based approximate string matching, or ASM, to accelerate multiple steps of the genome sequence analysis pipeline. As Onur also introduced, we focus on approximate string matching in this work because it is one of the core operations, or let's say core bottlenecks of the genome sequence analysis pipeline. And third, we implement an FPGA-based prototype for GenAsm, which is an ongoing work, and we call it BitMac. Here, we exploit the state-of-the-art 3D stack memory, HBM2, which offers high memory bandwidth. And also we exploit FPGA resources, which offer high parallelism by instantiating multiple copies of the GenAsm accelerators. And in the fourth work, which we call as SIGRAM, here we propose the first hardware acceleration framework for sequence to graph mapping and alignment problems. So SIGRAM is enabling efficient mapping of a sequence genome to a graph-based reference instead of a linear single reference like we did in GenAsm, for example, which provides more comprehensive and accurate genome sequence analysis. And here, like compared to a single linear reference, genome graphs provide a better representation of the diversity among populations by encoding variations across individuals in a graph data structure, which avoids a bias towards any single reference. So that's why in SIGRAM, we focus on this new problem. And let's start with the first work. And in order to understand this first work, we have to go one more level deeper and understand what are the types of genome sequence analysis that we can do. And there are two common big ways of doing this. The first one is called read mapping. And in read mapping, we align the reads against a reference genome to detect sequencing errors and also variations in the sequence genome. So this is one type of performing sequence analysis. And the second one is called de novo assembly. Here, we are combining the reads to construct the original sequence without a help of a reference genome. So why we would want to do that? We might want to generate the reference genome. So we may not have it. So we may want to come up with the first reference genome. That's one reason of performing de novo assembly. The other reason, we might not know the origin, like the actual organism that the sequence reads are from. So in this scenario, we cannot talk about a reference genome, or we may want to improve an existing reference genome. In that sense, we again perform de novo assembly. So these are two different types of genome sequence analysis, actually. And in this work, we will focus on de novo assembly because with the emergence of long-read sequencing technologies like PacBio or Oxford Nanopore, de novo assembly becomes a promising way of constructing the original genome, because a long read can span an entire repetitive sequence and enable continuous and complete assembly. In short reads, we did not have that advantage because they were short, so they could not cover repetitive sequences of the genome, which we have a lot, for example, in human genome. So they were not providing continuous and complete assemblies, but long reads changed this. So that's why de novo assembly became again a popular genome sequence analysis type, actually.
Damla?
Yes, Noor.
I want to ask here about the assembly method. What are the factors needed for a good, or let's say high quality assembly?
So that's a good question. One thing, for example, is coverage. By coverage, what I mean is, what is the, think about a original sequence, we have, let's say 3 billion positions, right? The number of reads that map or that is assigned to each location is called coverage, meaning that when we have higher coverage, it would it will mean more computation, but eventually it would help us to provide better quality assemblies because we have more reads that's covering each region. That's one thing. The other thing is the length, as I just said. So we have a lot of repeats in the human reference genome. For completeness, for be able to cover those, reveal those repeats, if we have a long enough read that covers that repeat, then it would mean that we can resolve that repetitive sequence portion of the genome. That's another important thing, for example. And of course, how long reads are helping is with fewer number of reads, you can cover a larger portion of the reference genome. So it's providing completeness in that sense. And one thing, maybe it's a bit detailed, in assembly, one of the metrics that we care is the number of contexts, meaning that number of full portions that we provide out of the de novo assembly pipeline. We would want to decrease that. And that's another, because we don't want to have a lot of fragments out of the full. We want to be able to merge them and come up with as full as possible outcome. So that's another metric, for example. So these are all important things to keep, and short reads cannot provide them.
So then based on your answer, then the read type can affect the result, right? What about the other species? Mm-hmm.
Of course.
So it also depends on the other species, right?
Yeah, exactly. So one thing which is being done in assembly, we did not analyze those type of pipelines, but one trend is also combining both short reads and long reads and performing assembly with that. Or since short reads are accurate, let's say, one of the main problems of long reads, as I said, is they're noisy. So some pipelines are making use of short reads, which are accurate, to like fix, to like correct or polish in this scenario, polish those like noisy portions. So it's kind of like there are multiple ways of achieving, but of course cost is increasing when you are merging two types of different like sequence reads from different technologies. So you wouldn't want to do that at the end, because it would mean that like performing sequencing for the same sample twice in the two different instruments, let's say. So it's complicated.
Yeah, thank you, Damla. So currently, what are the widely used assembly tools?
So that's a really good question. I think what we did as part of this, as I said, this was 3 years ago. So there are a lot of new tools emerged after we did this analysis, but there is not a single tool that's being used. There are multiples of them because of different reasons. For example, one is very slow but highly accurate, but another one is slow, fast but not accurate. So you need to do more on top of that. But Canoe, I think, which we did the analysis with, is still one of the state of the arts. But I know that there are more than that. Like I know Fly, I know some others, but I'm not very on top of what the actual current state is, actually. But there are a lot, and there's no single consensus about, okay, this is the way to go. Like we have kind of that consensus in read mapping scenario, for example. There are one or two to-go tools when you want to do like mapping, but it's not the case in the assembly pipelines. There are multiples which provide like let's say one of them is resolving repeats better. On the other hand, one of them is better for initial analysis. One of them is good for plant genomes. One of them is good for much shorter, much smaller genomes. So there's no consensus in that sense. I know that, but I don't know for what each category, what is the to-go tool for that. So it's a much bigger domain. And as I said, it's a very like fast growing domain compared to read mapping. So there are a lot of tools. So
yeah, it's clear enough. Thank you so much.
Yeah, of course. Thank you. All right. Is there any other question? Okay. So let's look at the genome assembly pipeline. So it starts with here in this scenario, the raw signal data, because in this work, we focused on nanopore sequencing technology, like long reads generated out of ONT devices. However, the only nanopore specific step that I will be explaining as part of the pipeline is this initial step, which is called base calling. Here, the raw signal data, which is the output of the nanopore devices that represent changes in electric current when a DNA strand passes through a nanopore. So we are starting with this raw signal data and converting it into our conventional DNA reads, which are composed of ACGTs, basically. So this step is called base calling. And as I said, this is the only step that is unique for nanopore sequencing. The rest is applicable. The rest of the steps that I will explain next is applicable to any long read technology, actually. So let's, in the second step, which is called read-to-read overlap finding, what we are doing is we are computing all pairwise read alignments between each pair of reads. So we find the R overlaps for each pair of reads. Then in the third pipeline step, which is called genome assembly, we generate an overlap graph where each node denotes a read. And if two like reads have an overlap between them, then there is an edge that's connecting those two nodes corresponding to those two reads, basically. We generate this overlap graph, and then we traverse it and construct the draft assembly by looking at the paths that we traverse in this overlap graph. And we come up with a draft assembly. And why we call it draft? Because it can be polished further with two other steps, two optional other steps, which I will explain next. So you can either leave it there if you are satisfied with the quality of the assembly, but if you want to polish it more and increase the outcome, of like the accuracy of the outcome, then you can apply these two optional steps, which the first one is read mapping. So here what you are doing is taking this draft assembly and taking the DNA reads from the first step, and basically you map the reads to this draft assembly. And then by using this mapping information, you perform like the last step, which is called polishing. Here, you basically polish the assembly with the help of mappings from the previous step. And as an outcome, you can get an improved assembly or consensus. So there are multiple terms used for that. So yes.
Maybe this is the right time to take questions from YouTube, because I see one question relevant to the data you show for each step. So we have one question says, do you have any security concerns for genomic data, and how do you find or collect these data for your research?
Now, that's a really good question, where we have Nur also working on like privacy preserving, like sequence analysis actually. So of course, so this is definitely a concern. And of course, cloud like architectures, cloud providers are being involved sometimes as part of this analysis as well. So when you have that, you have to rely on your like agreement with that. But when you are doing this in your like actual own compute cluster or whatever, then you kind of like preserve it. But of course, there are a lot of techniques like computational techniques to preserve like encryptions and those kind of things, where Nur can add more maybe here. But there are like more computational ways of preserving the privacy of your data as well, like in the other domains too. But of course, it's a concern, definitely a concern here.
Yeah, I agree with you, Dan, but there are many mechanisms proposed in the last years and many studies for sharing the genomic data while having the privacy guarantees, or we call it the privacy preserving sharing for the genomic data using many techniques. For example, one of the widely used is differential privacy, and has a lot of application for different genomic application or different type of genomic data set. So yeah.
Yeah, thank you, Nur. Is there any follow-up question regarding this from YouTube?
The second one was, how do you get the data, basically? Or where do you get it from?
Oh, us or the pipeline itself? So probably I can answer both, maybe. So
yeah, that will be good.
Yeah, for us, for example, we are using publicly available data sets. So we did not, and we don't right now, we don't have a wet lab where we have the sequencing devices, so we can collect some samples and run them and gather the data. We don't have that mechanism. So for our own research purposes, we use like commonly used like public data sets for our own research. But for like labs and other like outside of our work, so sometimes there are like real patients, the hospitals or labs can deal with them, so the data can be like collected. So what I mean as data is, of course, as I explained in various first slides, maybe I can show it. Where is it? Here. It starts with sample collection, as I said, and we come to this phase after a lot of going on in between, starting with sample collection, blood, saliva, whatever, and bone like tissue. It can be a lot of things, and then it goes through like a wet lab preparation process where they collect they extract the DNA molecule out of it, and after that, in the wet lab process, it's being chopped into smaller fragments, and then it's fed to sequencing devices. What we are interested in this phase, the sequence reads or the data that the sequencing device is producing. We don't have any mechanism to deal with this part as our own research. So that's two like different scenarios, whichever you're interested in. But as I said, the sample can be some donors, some patients, a lot of like some candidates, whatever. So yeah, I hope this answers the question.
Yeah, Damla, I have another question other than the genomic data set. So here, if we return back to accelerating the work, if anyone wants to accelerate the assembly pipeline here, So which step do you think is the most expensive in the pipeline to start with?
That's a really good question. So it's, again, depends, because there are multiple tools at each step which do their things in a different fashion, meaning in terms of different metrics. One of them is accurate but slow. The other is fast but not accurate, let's say. For assembly, this is the case, for example. But based on our analysis, we found out that base calling and assembly are two highly costly steps of this pipeline that we analyzed. We analyzed it for E. coli only, E. coli data, which is pretty small compared to, like, let's say, human genome, but we don't expect to observe, we should expect our results to scale to larger genomes as well. I don't believe the comparison would differ, but based on our analysis, as I said, base calling and assembly were two expensive ones. But again, I'm highlighting this. Base calling is specific to nanopore. It does not exist in as an external pipeline step in PacBio, for example. They do these kind of steps, like pre-processing steps, internally as part of their instrument. So they directly start with, let's say, overlap finding, for example. But another thing I would want to highlight here is read-to-read overlap finding and read mapping internally, they use the exact same tools, for example, like read mapping or overlap finding. They all end up in approximate string matching, for example. And that's why, as an expert, I will be motivating why we pick that and accelerated ASM, for example. But compared to read mapping and overlap finding, the all other, I would include polishing as well. Those three ones are more niche areas, let me say, compared to read mapping. Read mapping is a well-studied problem right now, which means that when we accelerate those steps further, the bottleneck is being shifted towards the other steps of the pipeline more. So, and in terms of polishing, so when the quality of the sequence reads will increase, this is my speculation, that's my belief actually. The need for polishing step will be less and less, because what we are getting out of assembly will be high quality. So the polishing step will be needed less in the future, not right now, we still need them. So that's why as a future work, I would say base calling or assembly, mostly assembly, because it's more common among multiple types of reads, would be that would be my next research if I was still a PhD student, let's say that way.
I like that, thank you, Damla.
Sure.
We have a tool from our group on assembly polishing, right?
That's true, that's true. That's why I said we still need it. That's why we worked on it.
Yeah, John Pertina is here.
Yeah,
he's the main author of that tool. He's happy now. Let
Which
me post the link
on
YouTube.
Yeah, but I also agree with Damla in that sense. I think the importance of the polishing tools will maybe decrease in time or maybe basically the polishing tools will shape into something else, basically, rather than correcting the bases. Maybe it's going to be more like scaffolding, or basically finding the gaps and making longer context, let's say, rather than trying to fix the individual bases, right? So yeah, I think I agree with you, Damla. And I also have basically two questions if we get back to the discussion regarding the data and the usage of data. So you mentioned that none of the other companies provide raw signal data, but do they ever produce or provide raw data in any sense? And if that's the case, for example, the Illumina sequencing machine or the PacBio sequencing machines, and if they provide, do researchers store and use such raw data, and what will be the motivations behind using raw data if they are doing it?
Actually, that's a really good question, which opens up a completely new research direction. And it's an ongoing, it's not new anymore because people has already started working on that, which is for example, skipping base calling completely, which is time consuming, we know, and trying to do the other upcoming steps, let's say not this pipeline, but read mapping pipeline, let's say the other type of sequence analysis, performing mapping on the raw data, using that raw data and dealing with the signal data directly is one ongoing research direction. So it skips base calling, meaning less compute time required for one step, and skipping that, jumping to another step, doing computation on the raw data directly. So there are works, ongoing works regarding this. That might be a really top advantage of dealing with raw data. But I think I also know that the companies are trying to keep that step as part of their instruments, like software, whatever, and then perform those kind of pre-processing steps there and directly produce more commonly used data like DNA reads to the for the later steps, so that there are more tools. It's a much populated area, like the dealing with DNA reads, so that it will be like more compatible as well. So maybe that's the reason why they still do that, because it's kind of like a niche area and they want to keep it near instrument, the pre-processing, base calling, whatever you call it, those steps. But yeah, I see like dealing with raw data directly can be a huge advantage, because we directly decrease the computation time by skipping one step. And here, as like in the nanopore pipeline, as I said, base calling was one of the major time-consuming steps. So, and the works that I mentioned are for the nanopore data, nanopore raw signal data, that they are the newer publications actually. Yeah.
Great. Thank you, Damla.
Sure. Do you have any other question? You said 2 questions.
First one was actually do other sequencing machines also produce, let's say, raw data, and do researchers store them other than basically ONT technologies?
As far as I know, yes, they generate it, but they as I said, they convert it as part of their own instruments pipeline and produce directly conventional reads or those kind of data as the output. But of course, all of them has some mechanism, which it can be a signal data or any other image data, something like that. Of course, they all generate the initial data like that, but they, as far as I know, Nanopore is the only one who is providing the raw signal data as the actual output. I'm not aware of any others, actually.
Thank you, Damla.
Yeah, thank you. Any other question? All right, we continue then. Okay, we went over the genome assembly pipeline using long reads. So let's look at our contributions as part of this work. So as I said, here in this work, we analyze the tools in multiple dimensions, and those dimensions were accuracy, performance, memory usage, and scalability. And we reveal bottlenecks and trade-offs that different combinations of tools, but also different underlying systems lead to. So we use 2 different systems, for example. One we call desktop, which is a very small server, just 8 cores, I think, yeah, and smaller memory. On the other hand, the other side, we use a system which we call as BigMem, which had 80 cores and 1 terabyte of memory. So we ran these tools on real systems, which is our one of the other main contributions. This is the first study on bottleneck analysis of nanopore sequence analysis pipeline on real machines. So we directly ran the tools on real machines, and as I said, we use different underlying systems so we could test how these tools perform in different environments actually. And we provide guidelines for practitioners such that they can determine the appropriate tools and tool combinations that can satisfy their goals. And also we provide guidelines for tool developers such that they can make design choices to improve current tools or also future tools. So in the paper, we have 21 observations, but here I will highlight 3 key findings we made, and you can refer to the actual paper in order to have to look at those actual 21 observations. So first, laptops are becoming a popular platform for running genome assembly tools. And if you ask why, because the devices, the sequencing devices are becoming more portable. And for example, one of the devices of Nanopore is just USB sized, meaning it's extremely portable. So you can plug into your laptop and it's a good fit for in-field analysis. But your compute platform, let's say laptops, should be able to provide the environment that you can support your in-field analysis. But we are dealing with greater memory constraints, lower computational power, and limited battery life, as you can imagine, in laptops. So we have to think about it, keep this into mind, and consider this when developing new tools. So that's one of our findings. The other one is, it's not a shock, but it's very important. Memory usage is an important factor which greatly affects the performance and the usability of the tool. There were some tools which we could not run on the desktop server that I mentioned, for example, because we were getting out of memory errors, so we could not run it. It's a huge issue because it's not the case that everyone has those large servers which they cannot afford we cannot afford those kind of servers all the time. So it's important. So data structure choices that increase the memory requirements and algorithms that are not cache efficient or not keeping memory usage in check with the number of threads were the problems that we observed as part of our this work actually. And the third one is, again, very important. Scalability of the tool with the number of cores is a very important requirement. However, which will come to our second finding, parallelizing the tool can also increase the memory usage. And based on our observations, we saw that not dividing the input data into batches or not limiting the memory usage for each thread, or dividing the data set instead of the computation between simultaneous threads were the issues that we observed as part of our analysis with those existing tools. And based on these findings, we reveal or motivate us that when we are designing tools or accelerators, which I will come next, are one of the goals we should have is achieving high performance while providing low power, so that, as I said, the limitations of laptops can be shielded actually. Or the second one is our designs should be memory efficient. And also the third one is scalability and high parallelism is of importance, so that when designing tools or accelerators, we should keep those 3 goals in mind in this domain, actually. Not just in this domain, but yeah, these are very important for genome sequence analysis. And as I said, for all other observations, we can refer to the paper. So in light of those 3 goals that we just discussed, we are saying that we are motivated that we need to co-design hardware and software. Just tool level design might not be enough in order to achieve all those 3 goals. To this end, in our second work, GenAsm, we are proposing a hardware software co-design for approximate string matching, which is one of the core underlying algorithms that's contributing to the compute bottleneck of genome sequence analysis. So just to recall, as I said, there are 2 ways. We covered de novo assembly as part of the de novo assembly pipeline as part of the previous work. But in read mapping, if you recall, read mapping was part of the de novo assembly. Besides, it's as a standalone, another and very common way of performing genome sequence analysis. And one of the very common downstream analysis of the read mapping is variant calling, for example. In order to perform variant calling, you have to perform read mapping. So and variant calling is kind of like the end goal of genome sequence analysis. So that's why read mapping is done a lot of times. So it is, and as I said, as part of the de novo assembly, it's also one of the major steps. So that's why in this work, we focus on read mapping. And let's look at the read mapping pipeline like we did for the de novo assembly pipeline. So it can be seen as a 4-step process. It is sometimes called seed and extend as well. And I explain why. So first, we are starting with a reference genome. If you remember, we have a reference genome and we are aligning reads with respect to this reference genome. And as the initial step, and it's also a pre-processing step, and it's only done once for each reference, actually, it is indexing. So basically, we are generating an index that contains all possible fixed length substrings as the keys in this index, and their exact match locations, those fixed length substrings, exact match locations in the reference genome as values. So we generate this hash table based index, and the actual core steps of the pipeline are these remaining 3 steps. As I said, indexing is done only once for each reference, and you can reuse the hash table for generated out of that reference, basically. So in the second one, what we are doing is we are getting the query read, and then we are dividing this read out of those fixed length substrings, which we call seeds. And after gathering these seeds out of the read, we are querying the index to find the potential mapping location. So we are looking at the exact match locations of each of these seeds in the index, and they constitute our potential mapping locations. But they can be a lot. And because of that, in the third step, which is an optional step actually, but it's Mohammed has a lot of works in this step, which show that it is very, very useful to perform pre-alignment filtering so that we can decrease the burden on the last, which is most expensive step of the pipeline, read alignment. Here, we are using filtering heuristics to examine the similarity between every read and its potential matching segment on the reference genome that are identified in the second step, basically. And here, the goal is eliminate most of the dissimilar sequences that we are 100% sure that they are dissimilar, so we can discard them and decrease the number of required alignments, which is the fourth step. So we perform the filtering and just report the remaining potential mapping locations to the last step. And for those remaining reference segments and query reads, we perform read alignment, or also it's called seed extension, where we perform a dynamic programming based alignment to determine the best location of the query read. And as part of this step, we also perform traceback between the reference segment and the query read in order to find the optimal alignment. So is there any question here?
Yeah, Damla, I have two actually. So the first one for the indexing step, how to choose the length and the location of the substrings for the reference indexing?
So that's a good question. There are some empirical analysis that has already been done for that, for sensitivity and those kind of reasons. So there are, for example, one of the very commonly used state-of-the-art read mapping tool, Minimap2, are providing those suggestions for, let's say, if you are using short reads, like Illumina reads, use these numbers because we empirically tested it, and this is providing the most accurate or sensitive outcome of it. Or for PacBio, do this, or for Oxford Nanopore, do that. So there are some analysis-based numbers that are commonly used in the field, actually.
So yeah, thank you for that. My second question again now here for the read mapping pipeline. Which step makes the read mapper so which is the most expensive step?
Read alignment. It is for sure read alignment. So it is like more than, of course, it depends on the input and everything, but around 70% of the full pipeline goes towards read alignment. So that's why we will be focusing on read alignment as part of GNASM, actually. Yeah, it is, and it's. I will come to the details, but the dynamic programming algorithm, like based algorithm, it is performing is dependent on the length of reference segment and query. So it's a quadratic time and space complexity algorithm. And if you think about, let's say, long reads, they are long, so they are contributing a lot to that complexity, which is making it even slower when it comes to long reads, for example. But anyways, it is the most expensive step regardless of the type of the read, actually.
Yeah, thank you, Damla.
Thank you. Any other question? All right. Okay. So just to provide some overview about read mapping, as I said, this is one of the first key steps in genome sequence analysis. And here we take each read, align it to one or more possible locations within the reference genome, and find the matches and also differences between the read and the reference genome segment at that location. As I said, when introducing read mapping, the next step after read mapping is variant calling, which uses these matches and differences in order to reveal the variations, like genomic variations that might exist in the sequence genome. So that's why it's a very core operation to perform read mapping. It's so important. And it is being done multiple times, as I said. It's a very common pipeline. And here, as you can imagine, we are performing alignment, and we are kind of doing string matching. But since we are also allowing differences, it is approximate string matching. And multiple steps of read mapping thus need approximate string matching, or ASM, so that we can account for sequencing errors and also genetic variations in the reads. So we cannot talk about exact string matching because these types of errors and variations exist. So we have to tolerate them while performing alignment or mapping. But this is one of the core main bottlenecks of the pipeline. And as we said, the bottleneck is computational power and memory bandwidth limitations of existing systems, and ASM is one of those major bottlenecks that is making GSA, which is genome sequence analysis, slow, basically. So to this end, our goal in this work is to accelerate approximate string matching by designing a fast and flexible framework so that we can utilize this accelerator for multiple steps of the pipeline. And also one of the goals here is to provide a framework for both short and long reads, actually. And to this end, we propose GNASM, and we base GNASM on BiTAP, which is an ASM algorithm that uses only fast and simple bitwise operations, making it amenable to efficient hardware acceleration. And GNASM is the first work that utilizes BiTAP for enhances and accelerates BiTAP and uses it as part of the GSA pipeline, actually. And this is also first ASM acceleration framework for GSA as well. And we modified BiTAP to support long reads and also to enable parallelization. And we also developed a novel BiTAP compatible algorithm for traceback. And we co-design specialized hardware for both modified BiTAP algorithm and also novel traceback algorithm, which is part of the under the umbrella of GNASM, actually. And we have, as I said, accelerators for both of these algorithms as part of GNASM.
So Damla?
Yes.
I like the way you explain about approximate string matching, and you have one reason why we need it, because there are sequencing errors and genetic variation. But as any other algorithm, can't we use something cheaper, computationally cheaper, where we can we don't need always the optimal solution, but we can use something really fast and simple enough for each application individually? Why we need such really expensive algorithms for these applications?
That's a really good question. So yes, you can apply those kind of maybe heuristic ways or more naive or cheap, as you said, algorithms, but eventually you need accuracy out of these algorithms at the end for the final product, because as I said, you will use that information for the next step, which is variant calling, and you don't want to miss anything or label something wrong, because you prefer to use a cheaper algorithm so that it can be fast. Yes, we want fast, but we don't want to lose any accuracy as the outcome of this final read alignment step. We can tolerate some for example, pre-alignment filtering. We are applying heuristics there, but we don't want to lose some meaningful information. We just want to discard the ones that we are sure that will not provide meaningful information to us at the end. We can tolerate that, but we cannot tolerate losing meaningful information at the end just to be fast and cheap, actually, because of the next step that will be coming out of after read mapping, which is top importance, actually, the end goal, basically.
How about something similar to Hamming distance, which is very cheap to be computed?
Mm-hmm. It cannot support indels, right? Which means deletions and insertions, which are very common very common variations that we see in, for example, structural variations, which are reasons of some diseases, let's say. So you cannot miss them. And in Hamming distance, it only supports substitutions because it's basically an XOR operation. It does not support indels. So you are missing that, which is incomplete, basically.
Mm-hmm. Yeah, I like that. So for the algorithm you mentioned, BiTAP, is this existing algorithm or your own implemented one?
It's an existing and very, very old algorithm, which John Alkan, which is our collaborator from Bilkent University, revealed or let us meet with that as part of their his algorithms, bioinformatics algorithms course. So he suggested that, as I also said, it only has very, very simple bitwise operations. So which we labeled as very amenable to hardware acceleration. So that's why we focused on this. When you look at the core algorithm, you would see that it is not you cannot say that, okay, this algorithm is efficient. It is an inefficient algorithm as it is. But as I said, we modify and extend it a lot so that it could be efficient and usable as part of the GSA pipeline. But it might be another motivation for people in our group or other researchers in other groups to look at those very, very old algorithms, or maybe not, it doesn't have to be old, but this is not a commonly used algorithm for sure. There might be, and there should be others somewhere outside, which we can just need to find them, reveal them, and modify them, maybe like we did here, and make them efficient and introduce to the field so they can change what we know for years, maybe. Yeah,
it's amazing to see something very old and still works, or at least you can build on top of it. This is algorithm is about 30 years old. And
yeah, that's true.
For the other algorithm, like Smith-Waterman, it's about 50, I think, around 50 years old, right?
Yeah. And Smith-Waterman and all those Levenstein distance, Needleman-Wunsch, they are, as you said, old, commonly used, accepted tools. I'm not saying we should replace them. I'm saying there are others out there which we are not aware of that might provide better solutions. Yeah, that's what I'm suggesting here, actually. We should just look at them. We should be open-minded. Let me put it that way. Another advice, which we learned out of our GNASM journey, actually, I think.
I love that advice, Damla.
Yeah, good. Thank you.
This is what Yale Pat says, right? Look backward. We need to look at these very old algorithms.
Yeah.
Old is
and there
gold,
are a lot.
Yeah, old is gold.
Yeah.
Yeah, that was great, Damla. Thanks.
Yeah, thank you. Thank you. All right. Any other question? Perfect. All right. So we are mentioning approximate string matching and BiTAP, but let's look at them in detail. So as I introduced, and as we said, the reads, we don't expect the reads to map like exactly because of genetic variations and sequencing errors. And here I will show what types of like errors we should be tolerating as part of approximate string matching in this use case. So one of them is deletion, meaning a character that exists in the reference, a character or a chunk of characters exists in reference may disappear in the read. Like a character or base appears in the reference might be substituted, switched to another one in the read, or a character or a chunk of characters that does not exist in the reference may appear in the read, which we call as insertions. So we have to be able to provide support for all these three types of errors as part of our approximate string matching algorithm. And as ASM, what it does is it detects these differences and also similarities between two sequences. So whatever I did not like highlight in this example are, as you can see, they are the matches basically. And in genomics, ASM is required first to determine the minimum number of edits between two genomic sequences, which is also called edit distance, and also to find the optimal alignment with a traceback. And here we find the sequence of matches, substitutions, insertions, and deletions along with their position. So traceback is required in order to provide this output as part of the read alignment or read mapping output. So what I mean, let's look at our example. What is the traceback output for this example is, okay, we have 3 matches followed by a deletion, another 6 matches followed by a substitution, and this new or like character is C. We should say that as well. And 6 more matches and a T is inserted and followed by 2 matches. So this is the traceback output we expect out of the approximate string matching algorithm, which will be used as part of the read alignment step of read mapping. And this approximate string matching is usually like commonly implemented as a dynamic programming based algorithm, like Smith-Waterman or Needleman-Wunsch, as we said, based on the like the actual use case. We use either of them. However, they are, as I said, they are commonly used, but they have quadratic time and space complexity. And because of that, it is desirable to find lower complexity algorithms for ASM, because this is problematic when the read length, for example, is increasing. So one candidate to replace DP-based algorithms for ASM is the BiTAP algorithm. And BiTAP uses only fast and simple bitwise operations to perform ASM, which I will show, which makes it amenable to efficient hardware acceleration. So since it is an ASM algorithm, it tackles the problem of computing the minimum number of like errors or minimum edit distance between a reference text, like reference genome, and a query pattern, like a query read, with a maximum of k many errors. So you have to provide that maximum error number to the BiTAP algorithm, which is different than the dynamic programming based algorithms for ASM. And the algorithm has 2 steps. It starts with a pre-processing procedure. And here we generate 1 pattern bit mask for each character in the alphabet. And here our alphabet is ACGT. So we are generating 4 pattern bit masks for the query read. And here, these pattern bit masks help us to represent the query pattern in a binary format so that we can directly use it as part of our second step, which is composed of pure bitwise operation. So we need that binary conversion here actually. And in the second step, what we are doing, and this is the main step, we call it searching or edit distance calculation here. We examine each character one by one by using pattern bitmasks from the pre-processing step, status bit vectors, which are updated at each iteration, and hold the partial match information for the text characters examined so far, and also, as I said, bitwise operations. It's, as you will see now, the algorithm is composed of bitwise operations only. However, as I said, the core BiTAP algorithm has its own limitations that hinder its applicability and efficient hardware acceleration for genome sequence analysis. And we found 5 of those limitations. And the first of them is data dependency between large number of iterations. So this is a pseudocode of the algorithm. We won't go into details. I will just highlight why we like these limitations exist. So as I said, BiTAP iterates over each text character of the like input text as the outer loop, and also as an inner loop, it iterates over each possible edit distance. And here, as I said, we have a maximum of k many errors allowed. So 1 to k is the inner loop iteration numbers. Let's say we have a long noisy read. As I said, we are talking about thousands of lengths and 5 to 10% of error rate, which means that both of these loops need to iterate over very, very large numbers when it comes to long reads. So that's one problem, that limitation we observed. And on top of this, yes, we have a lot of iterations, but on top of that, we have 2-level data dependency in BiTAP. One of them is there's an outer loop, it's the outer loop data dependency when computing deletion, substitution, and match bit vectors, as they require these old R bit vectors. And those old R bit vectors were generated in the previous text iteration. So we have data dependency in outer loop. On top of that, we also have inner loop data dependency when computing the insertion bit vector. It requires RD-1, and it is coming from the previous inner loop iteration, which adds another level of dependency on top of the large number of iterations. And when we look at the second limitation, as it is in the BiTAP algorithm, there is no support for traceback. So it does not include any like step or any additional mechanism for optimal alignment identification. What I mean is we are generating these 4 separate bit vectors for each type of case, like 3 types of errors plus the match case. However, in the Bayes algorithm, they are not stored, and there is no mechanism to like traverse them or process them so that we can come up with the optimal alignment. And if I use the term intermediate bit vectors, I mean those 4 bit vectors that we generate in the inner loop. So in the Bayes algorithm, we don't store them, we don't process them, so that there is no traceback support. And there's one more algorithmic limitation, which is no support for long reads. So in BiTAP, the query length, so the read length, is limited by the word size of the machine running the algorithm, because each bit vector has a length equal to the length of the pattern. And we perform the bitwise operations on these bit vectors. So the algorithm does not support long reads where lengths are on the orders of thousands to millions of base pairs, because we are limited by the word size of the machine running the algorithm. So we are not supporting long reads. And these are the algorithmic limitations of the BiTAP algorithm. And even if we solve these ones, we find that we cannot extract significant performance benefits with algorithmic enhancements alone. For example, while BiTAP iterates over each character of the input text sequentially, we can enable text-level parallelism to improve its performance. What we can do, we can divide the text into different chunks, and if we can have overlap, between those chunks, then we are not losing any edge cases, and we can perform perform the algorithm on each of these chunks in parallel. However, the achievable level of parallelism is limited by the number of compute units that exist in the system that we are running it. So we would expect that a GPU, which has thousands of compute units, can overcome the limited compute parallelism issues that CPUs experience, for example. However, we find that a GPU implementation of the BiTAP algorithm suffers from the limited amount of memory bandwidth available for each GPU thread because of the data generated by each thread is larger than the dedicated memory area or bandwidth for each of them. And these cover the hardware related limitations. And in order to overcome these limitations and design an efficient accelerator, we find that we need to both modify and extend the BiTAP algorithm and also develop specialized hardware that can exploit the new opportunities that our algorithmic modifications provide. And also we need it so that we can balance the compute and the memory requirements of the algorithm. To this end, we propose GeneAsm, which is an ASM acceleration framework for genome sequence analysis, as I said. And as I said, this is the first ASM acceleration framework for GSA, and it's also first that uses, enhances, and accelerates BiTAP. And we make significant modifications to BiTAP in order to overcome those 5 limitations. First, we modify the BiTAP algorithm by extending it to support long reads and also by eliminating loop carried data dependencies to enable parallelization. And second, we develop a novel BiTAP compatible algorithm for traceback, which uses the information collected during ASM about the different types of errors to identify the optimal alignment of reads. And these cover the algorithmic contributions of GeneAsm. And third, we co-designed specialized low area and area efficient hardware for both algorithms, and this covers the hardware level contributions of GeneAsm. And is there any question before I start the hardware design of GeneAsm? All right, I assume now. Okay, so let's look at the hardware design of GeneAsm. Our co-design hardware consists of 2 components. First, GeneAsm DC, which provides hardware support to efficiently execute our GeneAsm DC algorithm to perform distance calculation. So this portion is basically the modified BiTAP algorithm for the distance calculation step of approximate string matching. And also we have GeneAsm TB, which provides hardware support to efficiently execute our novel GeneAsm TB algorithm to find the optimal alignment. And this is the new traceback algorithm, like BiTAP compatible traceback algorithm that we develop. So this is the hardware support for that, or hardware accelerator match for that algorithm, basically. And also GeneAsm has 2 types of SRAM buffers. First, DC SRAM, and the second, TB SRAMs, actually. And let's look at the high-level overview of GeneAsm. So the execution starts when the host CPU issues a task to GeneAsm with the reference and the query sequences locations. And then GeneAsm DC reads the corresponding reference, text region, and the query pattern from the memory and writes these to its dedicated SRAM, which we call DC SRAM. And here, in order to decrease the memory footprint of the algorithm, we follow a divide and conquer approach, where we computation for each window. All of the windows, can you hear me? Can you see me?
We lost you for seconds and then.
Am I good now?
Yes.
Okay, something happened to my phone. Okay. So I will start from scratch for this part. So here in GeneAsm, in order to decrease the memory footprint of the algorithm, we follow a divide and conquer approach where we divide the text and also the pattern into overlapping windows and perform the traceback computation for each window. And after all of the windows' partial traceback outputs are generated, we merge them to find a complete traceback output. And like we did in text-level parallelism, if we can have overlap between two consecutive windows, then we are covering the edge cases as well. So we are not losing information in those like edges of the windows, basically. So we maintain that by having overlap between windows. So that in order to perform this like divide and conquer approach, Genasm first divides the reference text and query pattern into multiple overlapping windows. And then for each subtext and subpattern, its Genasm DC searches the subpattern within the subtext and generates the bit vectors, like we observed in the BiTAP algorithm. And then, since we will need these bit vectors as part of the traceback operation, we write these bit vectors to these TB SRAMs, which are sitting right between, like in the middle of DC and TB. After DC writes all of the bit vectors for the current window, then Genasm TB starts reading the stored bit vectors from TB SRAMs in the reverse order and generates the Windows traceback output, which is just for the current window, basically. And once Genasm TB generates this output, we go back to the next window and repeat steps from 3 to 7 until all windows are completed. We merge the outputs and report back to the host CPU. So this is the overall execution of Genasm, including both DC and TB steps. And with the help of our specialized compute DNS and also on-chip SRAMs, we match the rate of computation with memory capacity and also bandwidth, achieve high performance and power efficiency, and also we can scale linearly in performance with the number of parallel compute units that we add to the system, because there is no communication required between different Genasm modules. They can be responsible for different reference query pairs, so they don't need to communicate between them. So that with the help of that, we can scale linearly in performance, basically. Is there any questions so far?
We have a question, but I think you will answer it soon. What kind of hardware are you targeting? Is it FPGA, GPU, application specific?
Good question. We will come to that, but Genasm is designed as ASIC. But the next work that I will explain, BitMac, is the FPGA prototype for this design, actually. But I will show also that as part of our results, we are like Genasm is very efficient in terms of area and power as well. So it can be ported anywhere in the system as too. So, but here we have ASIC design, and we will have an FPGA based design next as part of the next work. Okay, I'm continuing. So let's look at the Genasm DC's hardware design. So we implemented it as a linear cyclic systolic array-based accelerator using very simple and small logic components. And this systolic array-based approach helps us to maximize parallelism and also minimize memory bandwidth and memory footprint. So because we can like transfer data between multiple processing elements without need of writing them to an outside like memory source, actually. So when we look at the smallest units, like basic compute components, which we call as processing core, here we are computing the intermediate bit vectors, all those 4 bit vectors, along with their embedded version, which is RD, which we are using as for our algorithm internally, basically. And when we add the flip-flop based storage logic around processing core, we define a processing element, PE, and those flip-flops helps us to maintain the data dependency between different iterations. And multiple PEs are concatenated to define a processing block, basically. And this is where we obtain like parallelism by running multiple iterations of the BiTAP algorithm concurrently, basically. So we have DC SRAM attached to our processing block, which stores the reference text, the pattern bitmaps for the query, and also the intermediate data generated from PEs. And also we have TB SRAMs connected to each. So we have one TB SRAM connected to one processing element, basically. And they store the intermediate bit vectors generated by each PE of Genasm DC for later use by Genasm TB. And when we look at TB's hardware design, we again implemented using a very simple logic. So what it does is it first reads the bit vector from one of the TB SRAMs by looking at the computed address, then performs required bitwise comparisons. So it's just basic comparisons to find the traceback output for the current position. And then it computes the next TB address, TB SRAM address to read the new set of bit vectors. And this would depend on whichever like operation we select for the current iteration. So if we select match, the next address is different. If it is insertion, deletion, subs, it's all different. So but this is the only like logic required by the TB hardware actually. And after Genasm TB finds the complete traceback output, which is also called cigar string, it writes the output to main memory and completes its execution and waits for the next Windows next Windows bit vectors to be written to TB SRAMs and then start its execution again. All right, so this was the hardware design of DC and TB. Let's look at the use cases of Genasm. So in order to demonstrate, we demonstrate the efficiency and flexibility of Genasm acceleration framework by describing 3 use cases of approximate string matching in genome sequence analysis. So the first one is read alignment step, which was the last step, if you remember. The second one is pre-alignment filtering, the third step. So Genasm right now is like used for short reads, like pre-alignment filtering for short reads only. And here we can again perform the ASM algorithm and the accelerator dedicated for that in order to like find the edit distance between the regions and then eliminate if they are above a predefined threshold. So we can utilize Genasm like that for this step. And also, as I said, BiTAP by itself is a edit distance calculation algorithm, which means that Genasm is like, it is one of the very natural use cases of Genasm because it's built on top of BiTAP. And here with edit distance, we are measuring the similarity. So it's another similarity metric or distance metric for defining 2 different sequences. And here we can support any arbitrary length 2 sequences to perform edit distance calculation. And we believe the Genasm framework can be useful for many other use cases. And we discussed some of them briefly in our paper. One of them is, for example, read-to-read overlap finding, as I mentioned in as part of the first work. Same tools are being used for read-to-read overlap finding and read mapping, which means that Genasm, since it supports read mapping, it can support some steps of read-to-read overlap finding as well. And also, as a non-genomics use case, we show that generic text search is a very natural use case of Genasm as well. So, as I said, we can either discuss more about them or you can refer to the paper for having more information about them. Okay, so let's look at our evaluation of the Genasm design and framework. So we evaluate Genasm using a synthesis of our DC and TB accelerator data paths and also detailed simulation-based performance modeling, which we generated in-house actually. We evaluate 16 gigabytes of HMC-like 3D stack DRAM architecture with 32 volts. So as I said, as part of one of the answers to one of the questions, our accelerators can be ported anywhere in the system. But why we use 3D stacked DRAM architecture is in order to achieve high parallelism and lower power consumption, basically. We put both DC and TB and the associated SRAMs in the logic layer of 3D stacked memory. And I will show next that our hardware is highly efficient in terms of both area and power. So they are within the budget limits of the logic layer. So the goal here is utilizing, exploiting the divided memory structure of the HMC-like memory so that we can put one full Genasm accelerator to each volt. And here we have 32 volts, meaning that we can fit 32 accelerators in the logic layer, which they can work in parallel without any communication requirements, basically. But as I said, this is the only reason why we ported them there, but they can be ported anywhere in the system. And for the use cases we evaluate, we compare Genasm with state-of-the-art both software and also hardware baselines. So I won't go into details, but I will just show them like briefly here, and I'm happy to answer your questions as well about the evaluation methodology. This is for the first use case, and these are for the other two use cases, our methodology, basically. Okay, so, is there any questions?
Yeah, I have a general question, Damla. If you want to compare the accuracy of Genasm or let's say any tool with other aligners, usually what are the accuracy metrics you are using?
That's a really good question. When it comes to alignment or mapping, so typically what is being looked at is the mapping location. So, which is kind of like not very useful when it comes to Genasm, because Genasm, as it is only like for the read alignment use case I'm talking about, it gets the seed information directly from another tool. We don't affect it because it provides us the locations for read and reference. We just align them. So that one is irrelevant. But in general, for read mapping, that is one of the ones. So if this read is aligned to position X in reference, and if another tool aligns it to position Y in reference, whether they are close enough, let's say in 10%, 1%, there's a metric like that, if they are close enough so that we can call them as correct mappings. This is one. The other thing is alignment score, basically. So what I mean by score is, so for each error type, let's say insertion, deletion, or substitution, you can provide a number or error score, let's say, or penalty for each of them. And for match, you can give a positive score. The others can be negative. The one match can be positive. So after you perform your traceback operation, you can count the number of insertions, deletions, substitutions, and matches, and can come up by using the formula number of matches multiply by its score and the rest, and for the rest, and add them. You can come up with a score, alignment score. So you can compare those alignment scores as well. So there is not a single tool that's trying to do that. We tried to do it, but it's a bit tricky. You can also look at the cigar strings, which are the traceback outputs. But even so, there are some heuristics applied, meaning that if there are two same scores apply for an alignment, then you only pick one case, and there's no mechanism to pick one over other with a meaningful reasoning, basically. So those heuristics can change the cigar string very easily. So just looking at the string itself and matching them, and if they match or not, looking at that is not a very good way of doing it. That's why alignment score is more commonly used, basically. But those two are, when it comes to alignment or mapping, are the two metrics that's used for comparing two outputs from two different aligners or mappers.
This is very comprehensive and clear. Thanks, Dan.
Yeah, thank you. And it's a good point, because I would like to comment about the accuracy of Genasm as well, because you can ask, okay, the algorithm that you have is an edit distance calculation algorithm, whereas there are more sophisticated ways of doing it. For example, as I just explained, providing different scores for each type of error. They can differ. And also there is a model called a fine gap model, where you are so for indels, this applies. If you are opening or insertion or deletion, meaning that the first occurrence of an insertion, you give a higher penalty. But when you extend that insertion or deletion, meaning gap, then you are providing a less penalty, because when you open it, extending it is cheaper and more common and more meaningful biologically. You don't see a deletion or insertion just a standalone, but as a chunk. So you don't want to penalize each of the insertions in that chunk with the same amount, but you only want to penalize the opening of it higher and the rest is as a lower. This is called a fine gap model. So these are more sophisticated ways of doing it. So we don't have full support as part of GNASM to these kind of more sophisticated scoring schemes, but we played with the algorithm, some ordering, and you can find more information about in the paper. Actually, it's easier when you look at the pseudocode to detect those changes, so that we can provide partial support for those type of scoring schemes, basically. So in our own code, what we did is the user provides, and this is very easy to implement in the hardware environment as well, but like just to give you an idea for our software version. So when the user provides the penalty scores for each type, then we are changing the order of how we like trace the intermediate bit vectors, basically. Let's say if the substitutions are cheaper, then we are starting at looking at the substitution intermediate bit vector when performing TB to see if it is a hit. So those kind of small changes we are doing as part of our design so that we can provide partial support for these kind of more sophisticated schemas. Or for a fine gap model, for example, since the cheapest errors are gap extensions, either insertion or deletion, then we are first looking at those intermediate bit vectors, then move the rest. So we are doing an ordering when we are tracing the intermediate bit vectors, basically, so that we can provide support. It's not 100% support. There are some edge cases where we cannot provide full, like the actual output, optimal alignment, but we observe that we are providing alignments that are within, most of the time, more than 90% of the alignment score. So, but it's a good question because this is something I would like to share as well as part of GNASM. Thank you, Nur. Okay. So let's look at our key results of GNASM, and let's start with area and power. So we synthesize our DC and TB accelerator data paths with a 28 nanometer process. And both accelerators operate at 1 gigahertz. And we find that, if you remember, we had 32 GNASM accelerators in total in our complete design, one for each HMC volt. So total area overhead for 32 GNASM accelerators is 10.69 millimeters square, and total power consumption is 3.23 watts. And we observe that both area and power consumption of a single GNASM accelerator at a single volt is around 1% of the area and power consumption of a single Xeon CPU core. So it's GNASM has low area and power overheads. And when we look at our results for the first use case, here as software baselines, we compare GNASM with BWA-MEM, which is shown with like pink red bars, and also Minimap2, which is shown with purple bars. And both of these tools are commonly used state-of-the-art read mappers. And we find that GNASM achieves 648 times speed up over 12-thread run of BWA-MEM, and 116 times speed up over 12-thread run of Minimap2, which are both software baselines, while reducing power consumption by 34 times and 37 times respectively. And when we look at our comparison with Gacktov-Darwin, which is a state-of-the-art long-read alignment accelerator. So Darwin is a hardware accelerator for long-read mapping, and GACT is its component, which is responsible for alignment. We provide, like GNASM provides 3.9 times better throughput, like on average, when we look at several different types of read length, different read lengths, basically, and 6.6 times better throughput per unit area and 10.5 times better throughput per unit power compared to GACT of Darwin. And for these results were for short reads. And for short reads, compared to same software baselines, Genasm provide, again, significant speed up and reduction in comparison, similar to long reads. And also, as a hardware baseline, we compare Genasm with ScyllaX of Genex. Again, Genex is a short read mapping accelerator, and ScyllaX is responsible for the alignment portion of it. So we compare with ScyllaX, and Genasm provides 1.9 times better throughput while using 63% less logic area and also 82% less logic power compared to ScyllaX. And as I said, I want to highlight that these are logic area and logic power because ScyllaX only reports these as part of its paper. Let's look at our results for our second use case, which is pre-alignment filtering for short reads. Here we compare with the state-of-the-art short reads pre-alignment filters, SUJI, which is Mohammed is the first author of that work. So compared to SUJI, again, state-of-the-art FPGA-based filter, Genasm is more efficient in terms of both speed and power consumption, while significantly improving the accuracy of pre-alignment and filtering. And for more details, again, either we can talk about it or you can refer to the paper. And for our third use case, edit distance calculation, we compare Genasm with the software baseline, EDLib, which is a commonly used software for when it comes to edit distance calculation. So Genasm provides 2 to 4 orders of magnitude speed up while reducing power consumption by 2 orders of magnitude compared to EDLib. And also as a hardware baseline, we compare Genasm with ASAP, and we show that Genasm provides 9.3 to 400 times speed up while consuming 67 times less power. And before I summarize Genasm, here is a list of additional details that you can find in our paper. I mentioned some of them, but this is the full list that you can see. So to summarize, in this work, we focus on approximate string matching because it is one of the main bottlenecks of genome sequence analysis, and we propose Genasm, an approximate string matching or ASM acceleration framework to accelerate multiple steps of genome sequence analysis. And Genasm is the first work that enhances and accelerates BiTAP for ASM with genomic sequences. And in Genasm, we co-design our modified, scalable, and memory-efficient algorithms with low power and area-efficient hardware accelerators. And we evaluate 3 different use cases and show that Genasm is significantly more efficient for all the 3 use cases in terms of throughput and throughput per unit power than state-of-the-art software and also hardware baselines. And you can refer to our paper for more details, as I said. So before I start talking about BitMac, is there any questions about Genasm or anything I just explained so far?
I have a couple of questions, Damla.
Yeah, go ahead.
If you go back to 56 slide.
Mm-hmm.
Yeah, I know it's a very difficult question and need a lot of analysis to know, but I'm always interested in knowing the contribution of each source of improvement. So you mentioned there are 3 sources, proposing new algorithm and then choosing the right hardware, then technology node or technology level might affect over there. Mm-hmm. But how much do you think each of these improvement or source of improvement might lead to the speed up you get, the huge speed up you get compared even to ASIC, to the FPGA existing work and so on?
Sure, that's a really good question. And as you said, this is as part of the review process, we got these questions multiple times as well. So, and we decided to have a dedicated section at the end and do the actual math to come up with all of these chunks. So algorithm level is the most tricky one, because it depends on the error like rate, the read length, and everything. But just to give you an idea what is included as part of algorithm level, the one thing is the divide and conquer approach we are following, which is a major one. And also the other thing is like being able to process multiple pairs in parallel, which is kind of like minor. But if I remember the number correctly, for the algorithm level, the divide and conquer approach was providing around 5,000x, something like that, for long reads. I think we have the actual number, but these are based on the actual BiTAP algorithm. So our baseline is BiTAP, but because I already showed the improvements over the other baselines. But when we think about BiTAP as the baseline in terms of algorithm level, we are talking about for long reads that much speed up or that much improvement. So it is significant. That's why we always said that, and still saying it cannot be just solved by hardware or software alone. That's why we need cooperation between both sides. When we come to hardware level, our PE design is the key actually there. So the scalability or the amount of improvement depends on number of processing elements directly. That's why we are highlighting that we are scalable. And in our design, we selected 64 processing elements because in terms of area power, like the sweet spot was there. And that means that it translates to 64 times speed up or improvement when we come to hardware level. And in technology level, we have 32 volts. That's why we picked HMC kind of like memory, so that we can put one accelerator at each volt, no communication. They can execute in parallel. So that's the technology level 32 times, because we can process 32 pairs in parallel, basically. So that's the technology level improvement we are providing in GenAsm. This is our rough numbers. I think especially for the algorithm level, we have more details in the paper, which we can either look now or anyone can look at to the paper. So,
that's great. Yeah. You cover almost all aspects of it. Thank you.
Yeah.
So mentioning the review process, as you said, so I know people normally don't see the efforts you spent on this work, or how much you struggle with reviewing or answering the reviewers and so on. So they just saw the last bit of it, which is the fruit. So I want to ask you if there is any interesting story you want to share with us during the review process or rebuttal.
That's a good question. So I won't give the name of the conferences where we tried and couldn't achieve, but one of them, I think I found this interesting actually. So one of the papers, pioneering papers in this domain, published in that conference 1 year ago, and we sent this paper, one version of it, to the same conference next year, for example. I'm not sure if it's okay. Yeah, the same venue. Nothing, and the scopes are completely same. And that one, for example, we did not have even a chance of rebuttal, claiming that this is not within the context of this conference. So it is shocking, I think. I'm not sure if this is interesting, funny, annoying, whatever you can label it, but it's interesting. So these things happen. That's what I'm trying to say. But eventually we were all the co-authors, which is a huge author list you might notice in this paper, we're happy with the end product actually. Yes, we of course had these kind of very annoying, disappointing moments along the journey, which this is a very long project, by the way, around 1.5, maybe even 2 years, starting from the very initial idea. So but yeah, these things happen, but I think it's important to gather the useful information out of each round, embed it into the paper, and then move forward that's, I think, I cannot say that you can do this when you are right in the middle of it, but when you look back, you can see that if you can do this, then you're fine. But yeah,
at least all the efforts get paid off right now.
Exactly, eventually. Yeah, it's of course tiring to have these kind of long processes, but other than that, I think, yeah, that's one thing. But other than that, we got really, as I mentioned, for sources of improvements, for example, we got that question multiple times, which makes sense because we have a lot of design choices. So we decided to put a dedicated section for that and explain every bit of it, which was really helpful, and it, I think, strengthened the paper. So that was a very useful comment feedback we got, for example, or accuracy, like as Nur asked. That was one of the main questions or feedback we got. So we embedded another set of section under read alignment use case, for example, to explain every detail of how we are comparing it, what we are achieving, and what we are missing, so that we could be transparent, so the readers, users can be aware of all details, and also it might end up with future work as well. So which I have at the end of my talk, some future works which touches some of these, for example. So we got definitely very useful feedback, which strengthened the paper at the end. So I'm not saying the opposite for sure, but yeah, of course we had funny, annoying moments as well along the journey.
Yeah, thank you so much.
Thank you.
Maybe I'll ask one question also.
Yeah,
I guess before asking the question, I'll make a comment. So I think what you're saying is basically you need to be resilient against any comment that's directed at your work, right? Even though the comments may be quite unfair or misguided, depending on the context, right? And I mean, I will also mention that it's I think maybe the right word is sad. It's sad to see that some people just reject works because they do not find it fit for a conference. And I think that's very dangerous. People should be much more accepting of different approaches and different ideas and different topics. That's how science progresses. I find it quite anti-science if people reject works for the reason that, oh, this doesn't fit my conference. Yeah, that's how you can basically make the conference smaller and smaller and smaller and worse, let's say.
Correct. Yeah.
Okay. What my question was actually on the previous slide. Yeah, this is the work basically. Which is, well, you mentioned there are 4 other potential use cases, and I think it's important to maybe you discuss them a little bit, because clearly this is very useful for the use cases that you mentioned, but whenever an accelerator is generalized such that it can cover many more use cases, its cost is amortized, right? And it becomes much more interesting to build. So what are those use cases?
Yeah, of course. Yeah. So we discussed 4 other possible use cases, 3 genomics, 1 non-genomics as part of the paper. So besides these 3 ones that we have already analyzed and provided some results for it. So one of them is read-to-read overlap finding. As I said, inherently, it is very similar to what read alignment or read mapping is doing. So in read mapping, you have reference, you have shorter reads, you are mapping them against that, but you never map it to the full reference, instead some regions of it. So you have at the end, 2 similar length sequences that you are performing approximate string matching, basically. And in read-to-read overlap finding, again, for example, Minimap2, or I think, Minimap2 is one of the very common tools for that step, for example. In that scenario, you are aligning 2 reads to reveal if they share an overlap. Same logic, again, ASM. So that's why it is one of the very possible use cases of GNASM to do read-to-read overlap finding, basically. The second one is hash table-based indexing. Yes, it is not done that way conventionally right now, but when you think about it, what you are really doing, you are setting a fixed length for substrings, and you are storing the exact match locations of each of these seeds or substrings within the reference genome, basically. So what you can do, since as the underlying BiTAP algorithm is can be used as exact match in exact match mode as well. When you set k, that maximum error, to 0, then it's an exact match finding or exact match finding algorithm, basically. So you can craft the algorithm in order to find those exact match locations of substrings in the reference genome to generate the hash table. One idea. The other idea is whole genome alignment, and we solved this in Darwin's evolution as well, for example. By Darwin, I mean the Darwin accelerator, not Darwin. So Darwin accelerator first published as a read mapping accelerator, then Darwin VGA is published for the whole genome alignment by minor changes to the architecture. So it is like in whole genome alignment, you have two large genomes, full genomes that you are trying to align with respect to each other. Again, approximate string matching, but in a very much larger context. But there are some, like we did in here, to divide this problem into smaller subproblems. A similar idea can be applied there, and GeneAsm can be useful for it. And these are 3 genomics based use cases. And the last one, I think, which can be very useful to what Onur said, like decreasing the costs, like NDA costs and those kind of things, like research costs. If you can find a use case that is even not genomics case, then it's even much useful. And here, generic text search, as you can imagine, is we are doing it every day, maybe, but we are just not aware that we are doing it. So like simple Google search, it's what we are doing, right? We are searching a text, and here we might miss a character, we might put additional character. So it's approximate string matching, basically. Maybe in not this easy level, but if you think about it, it's pretty much the same. The only difference is the alphabet size. Here we are dealing with 4 characters only, ACGT, but when you move to the generic text search, the alphabet is much higher depending on the language. But when we think about the algorithm itself, what is changing only is the pre-processing step, where we are generating 1 pattern bit mask for each character in the alphabet. So instead of 4, you are generating more, but it is it's a pre-processing step, and it's not part of that main body of the algorithm where all the limitations exist. So we are shielded from that part. We just need to generate more initial pattern bit masks and be done with it. So it's generic text search is a very natural use case of GeneAsm as well, because as I said, it's inherently a generic text search algorithm as well. But just we crafted it for genomics use cases here for analysis.
Yeah, thanks. That makes sense. So these could be also good avenues for potential future work if people are interested. And
definitely,
certainly text search has some challenges, right? You need to do some pre-processing to enable the accelerator to work, or you may need to sometimes change the accelerator also potentially, to match the alphabet, right? If you want to. But it seems like there's good opportunity for future research also.
Of course, yeah. And I will also show another like 4th work where I cannot say it's use case of GeneAsm, but it's like a more general case where GeneAsm can be one small piece of it. So which is for sequence to graph mapping. We did not list it here because it's not really a use case, but how you can broaden the work for a more comprehensive problem or goal. So I will show that as the 4th work, basically.
Okay, sounds good. Thanks, Damla.
Thank you. Is there any other question? Okay, then. For a reason I cannot do slide. Okay. Here we go. Okay. If there is no other question regarding GeneAsm or other like anything related to so far, I will talk about BitMac now. Okay, So next, I will be talking about two of our ongoing works, actually. Yes, I graduated, but there are still two of them, which I'm still working on, actually. So we build upon GeneAsm for both of them. And in the first one, BitMAC is where we propose the FPGA-based near memory acceleration or bit vector-based sequence alignment. And as you can understand, by bit vector-based sequence alignment, we mean BiTAP or GeneAsm. So it is basically the FPGA-based prototype of GeneAsm. But of course, it comes with its own challenges. It's not that easy to port an ASIC-based design to an FPGA-based design. So here we, in this work, we propose BitMAC, where we leverage a modern FPGA with high bandwidth memory on it for presenting an FPGA-based prototype for our GeneAsm accelerators. And our goal is to map GeneAsm accelerators to this specific FPGA, where HBM2 offers high memory bandwidth and FPGA resources offer high parallelism, so that we can, by instantiating multiple copies of the GeneAsm accelerators. So if you remember, in GeneAsm, we use an HMC-like memory, which was another type of 3D stacked memory. Here we have a real architecture which has another type of 3D stacked memory, which is HBM2 on it. So we can directly come up with our own prototype with all these components, actually. In order to achieve that, first we remodify the GeneAsm algorithms for a better mapping to the FPGA resources. And second, we exploit intra-level parallelism by instantiating multiple processing elements, PEs, for the DC execution. And here, for this step, it's kind of like together with the first one, because in order for a better mapping, for better utilization of the FPGA resources, we had to tweak with especially the design parameters like number of PEs or the size of the processing size of width of each PE, those kind of parameters for a better mapping. And also in order to exploit, use all the resources we have on the chip, we also introduce inter-level parallelism by running multiple independent GeneAsm executions, in parallel concurrently. So I will go over the key findings we have in the ongoing paper, basically. So when I say the complete BitMAC design based on the FPGA resources, what I mean is we have 4 BitMAC accelerators connected to each pseudo channel of HBM. So what I mean by pseudo channel is the memory, the HBM is divided into 16 pseudo channels or 8 channels. And we have 4 of these HBM memories. So 2 of them, so that in total we can come up with 128 in total, 16 multiplied by 2 multiplied by 2, basically, because we have 2 HBMs. And when we think about one BitMAC accelerator, what we mean is it contains a DC accelerator. Instead of 64, for better utilization, we increase that number to 16, a TB accelerator, an FSM design, and M20Ks. So what these M20Ks are, the on-chip memories that the specific FPGA has. So all the M20Ks are corresponding to the DC SRAM and TB SRAMs we have in our original GeneAsm design, basically. And we use, based on our place and route and synthesis and place and route, we clock the BitMAC design at 200 megahertz. And besides of course, because we are changing these parameters and the design is kind of like a new, we had to modify our spreadsheet-based analytical model for GeneAsm to fit our new BitMAC design. And we also report our performance results based on this analytical model. And we perform our BitMAC evaluation for the read alignment use case, the first use case from the GeneAsm paper, and with the same baselines, the read alignment steps of Minimap2 and BWA MEM as the CPU baselines. And also in the paper, we have GASL2 as a GPU-based baseline for GeneAsm as well, And so we included that comparison in our bitmap comparisons as well. And as you can see here, and as I said, these are like analytical model based results. We are observing, again, huge number of speed ups, like threefold over the CPU baseline, and around 7x to 19x speed up for the GPU baseline. So this is what we achieved by like changing our design. So this is more numerical analysis we had. So the actual like findings, like what are the problems, what was the challenges, let's look at those. So we observed that, as you can see here, we cannot fully utilize the logic units we have on the chip because we are limited. So our bottleneck is becoming the on-chip memory we have, basically. So we are, even though we find that there's a linear scaling of the FPGA resources with the number of instances we include in our design, due to high amount of data that needs to be stored for TB execution. If you remember those TB SRAMs, we have to store them in on-chip memory. We are bottlenecked by the amount of chip memory we have. So that's why we cannot go further, and which means we have to change something in order to decrease the memory utilization, but this is the by far we could go. And similarly, when we look at the total power consumption, based on our total dissipation analysis, sorry, we find that 59% of total dissipation accounts for the M20Ks, like those on-chip memory components. And the rest are like combination cells, register cells, clock network, and the static power dissipation. So again, M20Ks are the main contributors in the on-chip power dissipation as well. So by looking at these findings, we can say that we are bottlenecked by the amount of on-chip memory we have. And because of this, we cannot scale more, and we cannot saturate the high bandwidth that multiple HBM2 stacks on the FPGA provide. So this is not the bandwidth for the main memory is not the problem, but the storage and bandwidth for the on-chip memory is the problem, and we cannot go further. What we need is either newer FPGA chips, which will provide a higher amount of on-chip memory capacity, or algorithm level modifications. Even with some of them, as I said, we could go this far to decrease the amount of data that needs to be stored in M20Ks. So these are the, as I said, this is more like an extension work out of GNASM, where we are trying to come up with prototype, but these were the outcomes and also findings that we end up. So one thing that I will connect to the other work is, another option here is, since we are only targeting read alignment here, but read alignment is only one step of the full pipeline, but putting multiple steps closer and closer will decrease the data transfer bottleneck, or the cost comes with that data transfer. So one another option here is adding more steps from the full pipeline to the chip, so that we can exploit even more resources that these FPGAs provide to us, which will come to my fourth and last work, where here we can utilize this remaining bandwidth by including another step of read mapping pipeline to our design to be more transparent seeding here. Because if you remember, in seeding, we were querying a data structure which was sitting in the memory, so that it is promising that we might exploit that bandwidth that we are being presented by this HBM. But also here in this work, as I glimpsed over in when answering owner's question, we are exploring a similar but different genomics problem, which is called sequence to graph mapping. So before I jump into SIGGRAPH, is there any questions?
I have a question about the bitmap.
Of course.
What are the fundamental differences between bitmap and GNASM? you mentioned some challenges about memory management and such. But are there any differences in terms of design, for example, or architecture, algorithm?
Actually, one thing which we are doing in BitMac to decrease the TB-SRAM like size required for each PE is, so in of course there's a trade-off there. So if you remember the 4-bit intermediate bit vectors, we had 4. And in Genome, by only storing 3 of them, we could survive and perform TB. But here, because substitution can be generated using the deletion bit vector, so we only need 3 of them. Here, we, instead of storing 3 of them, we are dropping 1 more so that substitution can be generated out of deletion. We are also storing match bit vector, but we are not storing the insertion bit vector and putting it on as the last case to check on our list, as I explained as while answering Noor's question about accuracy. So meaning that we are from kind of like from 3 to 2 intermediate bit vectors, which would mean like decrease in the TB-SRAM size, not significantly, but slightly, because all of the like conclusions are for anything we design, anything we tried here in BitMac work is we need more on-chip memory. That's the only limitation we have. When we have more, we still have a lot to utilize in the logic side. We could scale more, but we cannot, because so that's all of our efforts went into decreasing the amount of data that needs to be stored in TB-SRAMs in Genome world, M20Ks in BitMac world, same thing. So that was the main, that is the main design change we tried. But other than that, as I said, even though it's not a very significant change, we decreased the number of PEs per accelerator, again, with the same logic. Less PEs means less TB-SRAM required for each accelerator so that we can replicate more. Those kind of changes we tried, so.
That's great. So do you describe these are design changes or algorithmic changes?
So one is algorithmic change for sure, the dropping 1 bit vector, but the other one is hardware design change, I think. So, which is kind of related to another algorithmic limitation, algorithmic change as well. So to give more detail, I talk about windows, right, in Genome world, where we are looking like as part of our divide and conquer approach. Mm-hmm. And we set it as 64 in terms of length of window in Genome, and it's same for here as well. So what I mean is when looking at each window in Genome, we also set the error threshold as 64. So everything was 64 length for query, pattern, input text, and also error threshold, so that we could utilize all those 64 PEs in parallel, because it's kind of like equal to the number of errors you want to tolerate. So I'm saying it's more detailed explanation. But when you think about it, you don't expect to have 64 errors in a window where you already have 64 characters. So it doesn't make sense. So we did some like empirical analysis there and find a sweet spot where if we only support 16, let's say, errors, which is like 1/4 of the original one, we are still good. When we think about the error rates, it makes sense because we are dealing with 5 to 10%. We are already on like around 25% of the window size. So we are good, which means that when we have 16 as the error rate, error like threshold, then we can have 16 PEs that map to that, so that we don't need 64 PEs, but 16, we can survive with that. So that's another design choice, which is based on an algorithmic change, basically. So there are, again, a software, hardware, co-design exists there. These are all things we change as part of BitMac so that we can better utilize the FPGA resources, but there's still things that might be done so that we can decrease this more, maybe.
Yeah, definitely. Research never ends.
Yeah.
So
exactly.
Do you think these optimizations or improvement can be still applicable to GeneAsm?
Of course. But the only thing I would doubt is that dropping the third one, because if you drop it, then you cannot play with the order of the bit vector like checking so that you can provide partial support for different scoring schemes. So they are connected. So that's why I said there's a trade-off there. You wouldn't want to drop that one. But there are other, there are ongoing projects in-house as well, which is related to GeneAsm, where we are trying to play with the algorithm for better mapping to other architectures as well. And as part of those projects, of course, changing the algorithm, the things that we couldn't see when like we were working on GeneAsm are being revealed for sure. So this is that's one also can be called as an ongoing project or directions.
That's a great, yeah. Thank you.
Thank you. Any other question regarding bitmap? All right. Then let's jump into SIGRAM, which is our hardware acceleration framework for sequence-to-graph mapping and also alignment. So as you recall, in read mapping, we start with reference, like linear sequence of a single reference genome. However, this linear representation of the reference genome results with ignoring the variations that exist in a population, which we can call as genetic diversity, and introduce biases for the downstream analysis, like variant calling, for example. And to address these limitations, recently, graph-based representations of the genomes, which are called genome graphs, have gained attention. So what genome graphs are? Genome graphs include the reference genome together with genetic variations and provide a compact representation and also enable us to move away from aligning with a single reference genome towards using the sequence diversity. So what I mean is, let me explain by using an example. Let's say this is one reference we have. So we are starting this as our single node. But let's say we have another genome. Let's not call it reference, but like genome, let's say. So in the same population, but here, instead of T, we are observing a G in that position, let's say. So what we can do, we cannot align the same read, let's say, to these references separately, right? It would be too costly. And we don't need to. What we can do, we can divide the common portions of these references, have one path with an inner T node in between to represent the first one, and another path to represent the second one. So we, in a compact way, we managed to represent both of these references. Let's say we have another one. In this time, we have an additional T after that T in the first reference genome. So what we can do, we can have an additional alternative path between those T and ACGT nodes, which would represent this third reference. So we will start from ACG, go to T again, and then ACGT. So we managed to represent third one as well. Let's say we have a fourth one where that T is completely missing. So there's a deletion exists. So what we can do, we can have a direct path from ACE to the first node to last node. So we will skip all of the intermediate ones so that we can represent the fourth reference as well. We put all the shared ones as nodes. So that's how we are providing a compact representation basically. So this is a typical genome graph we are dealing with, for example. And so since traditional read mapping causes reference bias, like we had in GeneAsm, for example, that was the traditional read mapping that we explained the pipeline. So aligning sequences to graphs emerges as a newer field. and practical tools only start to emerge, where most of the existing tools are specialized for one purpose only, or one type of read, for example, only short or long, whatever. So, and on top of that, given the additional complexities and overheads of processing a genome graph instead of a linear reference genome, graph-based analysis exacerbates mappings that, like the traditional read mappings, bottlenecks even further. So, such as read to reference mapping, right? Where the problem we are tackling in GeneAsm. So the bottlenecks that we revealed there is even worse here when we are dealing with graph-based analysis. So hardware acceleration of sequence to graph mapping is very, very important, but it is an unexplored research problem. So there is no hardware acceleration work that's targeting this problem, which is nice for us. So we propose SIGRAM. And here, our goal is to design a high-performance, scalable, power and area-efficient hardware accelerators that alleviate bottlenecks in both seeding and alignment steps of sequence-to-graph mapping, with supports for both short and long reads. As I briefly mentioned while I was introducing this work, so seeding is an additional step that we are incorporating in our SIGRAM design here, so that we can utilize the memory bandwidth even more with this memory access heavy step, which is seeding. And also we won't need to transfer data between different steps of the pipeline that sits in multiple places, one in hardware, one in host, for example. Instead of that, when we have both of them next to each other, then we are decreasing the amount of required data movements, like data that needs to be moved. And as I said, we are, again, targeting support for both short and long reads here. And SIGRAM consists of two main components. The first one is MinSeed, which is the hardware component corresponds for this seeding part. And this is the first minimizer-based seeding hardware, actually. And the second component is called BitAlign. And this is the first sequence-to-graph alignment hardware based on modified GeneAsm algorithms and accelerators. So this is the portion where we built upon GeneAsm, but as I will explain next, we have to, as I said, right now we are not dealing with a single linear reference anymore. We have a graph, so we have to adapt our algorithm, hardware design so that we can provide support. And as I said, both of these components are first in their categories, including SIGRAM. It's also the first graph mapping accelerator. So let's look at the overview, and also execution flow of SIGRAM. Before SIGRAM execution starts, as the pre-processing steps, each chromosome's graph structure is generated. Then each graph's nodes are indexed, and both the resulting graph and hash table-based index are preloaded to the memory. As you can see on the upper side, we have the main memory, which is storing graph-based reference and also the index. And SIGRAM execution starts when the query read is streamed from the host CPU, and MinSeed, which is the first component responsible for the seeding part, writes it to the read scratchpad. And using all of the k-mers of the query read, and by k-mers I mean the k-length substrings. I did not mention the k-mer notation when I was explaining the read mapping pipeline, but when I say k-mer, it is the k-length substrings. So fixed size is k in this notation, basically. So using all of the k-mers from the query read, MinSeed finds the minimizers and writes them to the minimizer scratchpad. And for each minimizer, then MinSeed fetches its frequency from the hash table at the main memory, meaning that for that minimizer or for that picked k-mer, the number of occurrences, exact match occurrences within the graph-based reference. So that's what the frequency is. And then filters out the minimizers whose frequency is above a user-defined threshold. And this threshold is typically determined beforehand for each chromosome. So each chromosome structure, for example, Minimap2 has a fixed frequency. So it's not even a user-defined threshold, basically. It depends on the chromosome that we are performing the mapping against. And next, MinSeed fetches the seed locations of the remaining minimizers, that the non-filtered one, from the main memory and writes them to the seed scratchpads. And for each seed, MinSeed calculates the candidate reference region for each seed, meaning that the leftmost and rightmost locations in the reference that we need to perform the alignment against. So that is the all steps required as part of MinSeed execution. And after finding this, the reference region, we fetches the graph nodes from the memory for each candidate region that we calculated as part of step 6, and we write them to the input scratchpads. And that's where we have the reference region for the for performing the sequence to graph alignment step, where bit-align will be responsible for. And also MinSeed, of course, since this is input scratchpad, we need the query read as well. MinSeed sends the query read to bit-align as well. And then bit-align, once bit-align has its own inputs from MinSeed side, it starts generating the bit vectors required for the distance calculation step of the alignment. And this you should be familiar with because of the GNASM background you already have. So that after that, this time, bit-align writes these not only to TBSRAMs, but also hop queues in order to handle the hops required for the graph alignment. So as you can imagine here, we are not dealing with neighbor-like characters anymore only, because we might have hops coming from a non-neighbor character. So in order to handle that, writing the bit vectors to TBSRAMs alone is not enough. We have to be able to handle the hops, and for that reason, we need hop queues. So when we write all of these, bit-align finishes writing all the bit vectors. Then it performs a traceback, which is very similar to GNASM TB algorithm. So it just adapted for handling hops, as I said for the DC part, and reads the bit vectors from bit vector scratchpad, performs a traceback operation, and finds the optimal alignment between the subgraph and the query read. So the internals are very similar to GNASM, but there are, of course, significant changes that we need so that we can support the graph structure.
Damla, I have a question.
Yes. Go ahead.
So what are the challenges you have faced in designing bit-align? I mean, to modify GNASM into bit-align, especially for handling the hubs?
Yeah, sure. So that's a really, really good question, because it is one of the most challenging parts of the SIGRAM project. And I wouldn't want it to be observed as just a minor extension of GNASM, because it is not. So if you think about the linear reference, I wish I had a picture or something like that to show you here. I might have oops. Just give me a second, please. And this would make it clear to understand how DP-based algorithms you do this as well, I think. Just give me a second, please. Okay. You cannot see my screen anymore, right? Okay, you can see it, right?
Yes.
Okay.
Yes, go ahead. Dan.
So this is the linear version, let's say. I will explain it in DP world and then translate it into GNASM and SIGRAM or bit-align world. So here, when you think about the linear reference, in order to calculate this cell, what you need is, because you are only interested in this one, let's say, or this one, you are only looking at upper, left, and diagonal cells, because those are the only So if you think about edge, this is the only edge that this G has. But when you move this to a reference world, and I hope you can see my mouse as well. So,
yes.
Okay, good. So here for this G, for example, we have another edge coming from here, for example, meaning that all of the data here, we are interested in that one as well. So we cannot be limited by these three, but we have to fetch data from these two cells, which correspond to this node where we have an edge as well. So this is same for GNASM or bit-align. We cannot only have bit vectors coming from those previous iterations, like right 1- iterations, but we can have from any of the previous iterations as well. So what is challenging here is, again, memory size. So do we really want to? Yes, we are storing all of them anyways for performing TB, but we are storing them in TB SRAMs, right? Or bit vector scratchpads. Does it mean that each PE, I think right now, I can switch to my bit-align slides to explain that. Let me do it quickly. Okay, here we go. Okay, so this was, don't think about hop queue registers. This was the PE, let's say original PE or similar version of it from GNASM. We have TB SRAMs. But if we want to have data from non-neighbor PEs, it would mean that we should have this blue connection for each TB for all of the TB SRAMs that are connected to other PEs as well, which is a huge amount of overhead because a lot of connections are needed, a lot of maintenance and flow control are needed. In order to limit that, what we did, we put these hop queue registers, which are storing the most recent X amount of bit vectors for us. And they are being provided, as you can see here, to each PE from left-hand side and right-hand side, so that one of them will be providing old R, the other one D-1, the other one is old RD, which are details. But just to think about this, in order to get rid of these TB SRAM connections all over the PEs, we put these extra registers. But of course, it will come with another challenge. What will be the size of these hop queue registers? So how many of the most recent bit vectors you would want to store? So we did another empirical analysis to find that number, and we find that 12 is makes sense in order to cover most of the hops that exist in the graphs that we are dealing with as part of SIGGRAPH. So that's why we decide on 12. So each of these hop queue registers that you see, which are additional memory components, by the way. So we are adding more area and power to the system, but we have to, in order to limit the number of connections that TB SRAMs and PEs have. And these are like shift registers. So they are being fed with the most recent data, and the least recent data is being thrown out. And as I said, the queue length is 12 in our design based on our empirical analysis. This is the major challenge we had, and this is how we solved. Yes, it might sound like a very easy change, but I hope I was able to explain all from start to end. So it wasn't like all details, like we had to do extra analysis for each of them based on our inputs and design choices. Yeah.
Yeah, I think you did a good effort for that. It looks, of course, novel by solving all of these challenges.
Yeah. And here, one other thing which I can highlight here is these hop bits, for example, and those hop bits are responsible for telling that whether the current text that we are processing, which hops, with which characters it has hops with, so that it's a binary array you can think of. Again, same length with the hop queue registers, telling whether we would want to achieve the hop data from it's 1 or 0. If it is 1, it means that there's a hop between that current node and the node, whichever like position it has. So we are going to hop queue register for that specific position, fetching the data, like the bit vectors for that text character, and putting it in our PC like processing core to perform the logic with the bitwise operations, which are pretty similar to GNASM. So that's another detail that we had to include here. And pattern bit mask is the same with GNASM. And as you can, I'm not sure if you can remember, but we had 2 more bit flip-flops here in order to preserve the data dependency. They are gone here. We are maintaining everything with hop queue registers now. So that's, yeah. So the intermediate bit vectors are still going to TBS RAMs, but the actual old RD, the ended versions of them are only being handled with the hop queue registers. So that's how we are achieving the graph-based alignment.
Thanks, Damla. So as far as I understand here in SIGRAM, the bit align will execute the most time, right?
Correct, which I will explain more, but how we designed it is, so for example, let me see, okay, this is one slide before it, but here for MinSeed, we have several scratch pads, right? Which I already explained as part of our like overview for SIGRAM. For all of these scratch pads, we applied double buffering so that we can fetch more data than we need from main memory and store them beforehand, while bitalign is performing its execution for the current set, so that we are removing MinSeed completely out of the critical path of the full execution, and bitalign is the only responsible step or execution for the total like runtime, basically. And yes, it is definitely, even if we did not do this, even if MinSeed was on the critical path, bitalign is the major part of the full pipeline. It is significantly, it is a significant portion, yeah, for sure, yeah.
I see.
Even worse than what we observed in the regular traditional read mapping, where in that one, alignment was the bottleneck. We knew that, and as I said, in this world it is even worse. But yeah, but with putting those hop queues into the system, we are eliminating the latency introduced by handling hops. So with those additional components, we are still managing to do all computation in a single cycle for each iteration. So that's another trade-off. We are putting more memory components to the system, increasing the area, overhead, power consumption, but we are not losing any performance compared to the traditional read alignment scenario. That's another point I would, yeah,
that's a great, thanks.
Yeah, thank you, Noor. That was a great question. Any other question? All right. So I will quickly go over this. As I said, I have already covered all of this stuff as part of the overview, but in our MinSeed hardware, we have 3 computational modules for finding the minimizers, filtering the frequencies of minimizers, and finally finding the associated regions of every seed location. We have 3 scratch pads for storing the read, minimizers, and seed locations. And finally, we have a memory interface which handles the frequency, seed location, and subgraph accesses. So in bitalign hardware, which we have already covered, it is again similar to GNASM, a linear cyclic crystallic array-based accelerator on top of what we had in GNASM. Of course, besides all other changes, we have hop queue registers to incorporate the hops by feeding the bit vectors of non-neighbor characters and nodes and maintaining it knows additional latency on top of what GNASM had already had with the help of those hop-queue registers. And when we look at the overall system of SIGRAM, so we have one HBM2 stack, and in our full design, we have 4 of those stacks. And we have 8 channels in one HBM2 stack, and we connect one min seed to each of those channels, meaning that we have, in a single SIGRAM, we have 8 min seed modules that exploit data level parallelism and performance seeding. And we have, and they have, yes, exclusive access to one HBM2 channel, which is kind of like, which I implicitly said already. And as you can see from the figure here, we have one bit align module connected to one min seed module here. And we have already covered this, but we hide the latency of min seed when performing seeding while running sequence to graph alignment with bit align with help of the double buffering technique we apply to all of the scratch pads that the min seed has. So while it's sending the next alignment like pairs to bit align, it is fetching the other set from the memory, so it's not on the critical path. But anyways, like bit align is the most expensive in terms of like breakdown component anyways.
Damla?
Yes.
Another question, please.
Yeah, sure.
So here you are using the high bandwidth memory for SIGRAM, but can we achieve the benefits of SIGRAM without implementing it using the high bandwidth memory?
Definitely. It applied for GNASM, it applies for SIGRAM as well. As long as we can manage the memory accesses, so the only reason, again, we are using HBM to be able to exploit that channel level division it has, so that none of the min seed accesses among those 8 min modules communicate or like compete with each other. As long as we can come up with such a memory design or memory controller, whatever, we don't have to have an HBM here. And this might be one change we will be implementing in SIGRAM because it's ongoing anyways, because that's a worry we get or we might get actually. Mm-hmm. So that's one thing we would want to address for sure.
Yeah, it will be, I think, good to show that. Also, you mentioned here that you are supporting processing like multiple seeds, right, in parallel. Can SIGRAM also support processing multiple reads concurrently in parallel? I mean,
that's a good question. Yeah, as I said, we have 4 stacks of HBM, and they are completely independent of each other, and the content of each HBM stack is replicated. So they all are identical, meaning that we can like send one read to each of those stacks and have 4-level parallelism. So we can concurrently process 4 reads in parallel because with the current design, because we have 4 independent stacks in the system. But within each stack, so within each SIGRAM like module, we don't support multiple reads because a single read provides a lot of seeds for us to process, which is like making it occupied all the time anyways. So that's why. But other than that, there's no like direct support for multiple reads within a single SIGRAM accelerator, actually.
I see. Thank you, Damla.
Yeah, thank you. Okay, any other question? All right. So let's look at use cases of SIGRAM as we are shaping this as a framework as well, which would be saying that SIGRAM has multiple use cases. The first one is the natural one, end-to-end sequence graph mapping. So min seed plus bit align, the whole SIGRAM design will be executed, and it will be providing support for end-to-end sequence to graph mapping, basically. And here, since we are building upon GeneAsm and it has support for both short and long reads, same logic applies here. Cgram supports both as well. So another use case is sequence to graph alignment, meaning bit align can be executed by itself without the need of an initial seeding tool or accelerator. Like GeneAsm, it was a read alignment accelerator, and the seeds were being fed to it by a host CPU, by an outsourced tool. The same can happen here. Bit align doesn't need min seed. It can be like paired with any type of software or hardware seeding tool, actually, or accelerator. And one thing to note here, bit align can also be used for sequence to sequence alignment, like traditional read alignment, since sequence to sequence alignment is just a special and simpler variant of sequence to graph alignment. When you don't have any non-neighbor edges, like what we call as hops, when everything is linear, then you have a graph without any hops, which is the exact same thing what sequence to sequence alignment is doing. So the other way around is not correct. Of course, sequence to sequence alignment accelerators or tools cannot perform graph alignment, but whatever tool or accelerator, including bit align, that performs sequence to graph alignment can also be used for sequence to sequence alignment as well. And as a third use case, the seeding alone, like MinSeed, similar to bit align, MinSeed can be a standalone module for seeding, which can be coupled with any alignment accelerator or tool. So it is orthogonal for any of them. So, and the important thing here is there's nothing special in MinSeed that makes it specific for graph alignment or graph mapping, meaning that any sequence to sequence map as part of any pipeline for sequence to sequence mapping or sequence to graph mapping, MinSeed can be utilized. So that's another note for seeding, like MinSeed use case. Any questions?
Yeah, Damla, again. Yeah, is there any other potential use cases for Cgram? For example, metagenomics or such applications or other use cases?
Oh, no, I just managed to hear you. Sorry, I completely lost you.
No problem, no problem. I'm asking about the other potential use cases for Cgram. Can we use it, for example, for metagenomics?
So the thing is, how we can use Cgram, yes, of course, any use case that would involve mapping or alignment, including metagenomics use case, can be, of course, applicable for Cgram as well. So, we know that in metagenomics, we perform alignment multiple ways in different set of a lot of references, right? So if it would make sense, we can represent them in a graph format. So Cgram will be directly used for that. Or similar to GeneAsm, like a non-genomics use case, if we want to look for words in a dictionary of words to see if they match. Again, if that dictionary is represented as a graph, then it's another use case of Cgram in a non-genomics domain as well. So those are all possible use cases of Cgram for sure. And maybe we can think of more going forward as well.
That sounds very good. Thank you, Damla.
Thank you, Noor. All right, let me continue. So again, similar to GeneAsm, we evaluate Cgram using synthesized system Verilog models of MinSeed and bit align accelerator data paths. And also we have simulation and spreadsheet-based performance modeling. I explain the overall system. We have 4 HBM2 stacks, each with 8 independent channels, and 1 MinSeed and 1 bit align per each channel, meaning we have 32 Cgram accelerators in total. And for our baselines, we have Graph Aligner and VG for sequence to graph mapping use case, Pascal for sequence to graph alignment use case, which we are in the phase of expanding this, like with more baselines. And also we provide results for sequence to sequence alignment, because as I said, Betalign can perform sequence to sequence alignment so our baselines are Darwin, Genex, and Genasm for that. And we use simulated data sets for both short and long reads. And for the reference graph, we use the latest major release of the Human Genome Assembly as a starting reference genome. And for the variations to generate the graphs, we use 7 VCF files from the Genome in a Bottle project, actually. And so let's look at our key results and start with area and power. So again, based on our synthesis of MinSeed and Betalign accelerator databeds with a 28 nanometer process, when both of them run at 1 gigahertz, we find out the total area overhead of SIGRAM accelerators attached to all 32 channels is around 25 millimeters square, and the total power consumption is around 17 watts. And we find that main contributors for the area overhead and power consumption are HOPQ registers, since they constitute more than 60% of the area and power of Betalign DC logic, as you can see here. And also the bit vector scratch pads, which are here. So those two are the main contributors for the area overhead and also power consumption. And we also in this table show the breakdown for a single SIGRAM, for a single HBM stack, and for all the total system here. So let's start with our comparison of SIGRAM for short reads. And one of the baselines, as I said, is graph aligner. When we look at this analysis, we find that SIGRAM provides around 9 times throughput improvement over graph aligners 12-thread execution, while reducing the power consumption by around 5 times. And when we did the same analysis with the other baseline VG, we find that similarly 7 times throughput improvement over VG's 12-thread execution, while reducing the power consumption by 6.5 times. And when we do the same analysis this time with short reads, we observe higher throughput improvements over both graph aligners 12-thread execution and also VG's 12-thread execution. And also, when we look at the key results for the sequence to graph alignment module of SIGRAM, which is Betalign, first we compare it with Pascal for the sequence to graph alignment use case. And we find that based on the power, the paper numbers that Pascal reports, Betalign provides for different data sets around 40 times to 540 times speed up over the 48 thread AVX512 supported execution of Pascal. And as I said, Betalign can be used for sequence to sequence alignment or linear alignment as well. And for the cost here is because we are providing more functionality than needed in Betalign when we are just running linear alignment is the extra HOPQ registers. But as I said, as I highlighted, we don't sacrifice any performance to do that with the help of those extra registers we are putting in our system. And for our comparisons, we compared over Gacktov, Darwin, and Genasm for long reads, and also over Silax of Genex and again Genasm for short reads. And we use the optimum configuration of each accelerator reported in their corresponding papers and show that Betalign is providing throughput improvements over those baselines, but of course it comes with higher power consumption, higher area overhead cost because of the, as I said, the extra functionality we are providing here by supporting both sequence to sequence and sequence to graph alignments. So to summarize, since traditional read mapping causes reference bias, aligning sequences to graphs emerged as a newer field and practical tools only start to emerge. where most of the existing tools are specialized for one purpose. Given the additional complexities and overheads of processing a genome graph instead of a linear reference genome, graph-based analysis increases the analysis bottlenecks of read mapping. So hardware acceleration of sequence-to-graph mapping is an important but unexplored research problem. So in this work, we propose SIGRAM, the first hardware acceleration framework for sequence-to-graph mapping and also alignment. And we base SIGRAM on a memory-efficient minimizer-based seeding algorithm and a novel bit vector-based highly parallel sequence-to-graph alignment algorithm, which are min-seed and bit-align. And we co-design both of our algorithms with high performance and area and power efficient hardware accelerators and propose the first hardware accelerators for both. And we compare SIGRAM and bit-align with state-of-the-art sequence-to-graph mappers and also aligners. And we find that both provide significant speed up compared to the baselines while reducing the power consumption, except the case that when we perform sequence-to-sequence alignment with bit-align, because we were increasing the power consumption in that scenario compared to the sequence-to-sequence alignment accelerators because of the more functionality we provided. So, and with SIGRAM, we cover all the pieces that create my PhD work. And to conclude, rapid genome sequence analysis is bottlenecked by the computational power and memory bandwidth limitations of existing systems, as many of the steps in genome sequence analysis must process a large amount of data. However, as I show as part of this, my PhD journey, we can accelerate genome sequence analysis by co-designing fast and efficient algorithms along with scalable and energy efficient customized hardware accelerators for the key bottleneck steps of the pipeline. And to this end, I provided like sometimes overview, sometimes in-depth analysis, like walkthrough of 4 works, which like constitute my PhD dissertation. And before I will jump to future work that I list some of them, I would be happy to get any questions. And thank you.
That's great, Damla. So you have GenAsm, you have BitMac, and you have SIGRAM, right? And only BitMac was implemented for real FPGAs.
Correct.
So how about the other 2 works? Are they simulation based? Do we have real devices doing these operations, giving the importance of genome analysis for any of these accelerated steps?
So that's a really good question. So BitMac is already like related to GenAsm, almost same. So that's I think BitMac covers GenAsm somehow. But when it comes to like releases, we release the software version of GenAsm basically, so it's available in GitHub. And for SIGRAM, it might be a very nice future work or extension work out of SIGRAM actually. Yes, it's an ongoing work, but still I think it might deserve a separate like project as well. As I said, we have similar projects for GenAsm, so why not for SIGRAM? So that's definitely an option. And another note, which when you asked came to my mind, there's no end-to-end sequence-to-graph mapping work on GPUs, for example. There is one limited one for alignment portion only, but there's no work for GPUs for sequence-to-graph mapping. I did not list it on my future work, but came to my mind when you were asking your question. So that might be a good future direction for someone from our group or for other research groups, I don't know. So that's one thing I can add. But for FPGAs, the good thing is our implementations, it was same for GenAsm, it is same for SIGRAM, even though it is kind of like under NDA, some parts of it. So that's why we couldn't release the hardware code of GenAsm, for example. So for FPGAs, it might be easier to do the mapping. Not easier, but same effort like we did in BitMac for mapping Genome to an FPGA. Same for SIGRAM, actually. And even better, we have the seeding module implemented in hardware this time as well. So it might be another really good showcase to say that, okay, hey, we have this real product for SIGRAM as well, which is very comprehensive in terms of steps and the problem. And
but you won't achieve the same speed up as you got from ASIC, for example.
Yes, that's true. That's very true. You would want to have that. You wouldn't want to. It's a bit like extreme target to put, because ASIC you have more flexibility. You can throw more resources and be done with it. But yeah, that should be the goal for sure. Whatever.
How about other real devices where they provide processing near memory or processing inside memory, as what we got recently from Samsung or UpMem architectures?
Yeah. So that's another good question. So I think the challenge here, when I think about the algorithms itself, the underlying algorithms, is the shift that we discuss sometimes internally about that as well. Shift operation, bitwise shift operation is a very core operation that we need as part of GNASM, beta line, whatever you call them. So shifts might be problematic when we want to port these in like processing using memory technologies like Samsung, what Samsung provides. So, but it has to be taught thoroughly, actually. But that's the very initial challenge that comes to my mind when I think about using memory to those operations. Yeah. Other than that, they are very promising, but I'm not sure how GNASM or SIGRAM are good fits for those kind of architectures.
I see. Yeah, when we go for different architectures, so the main concern always is the configurability or the flexibility in choosing these parameters or choosing the algorithm, the data, and so on. So how flexible to rechange any of the parameters, input parameters, or the data, basically, to these three the main work you presented?
So in terms of input data, we are pretty flexible because of the divide and conquer approach. We are dividing whatever is provided to us, so we don't have any limitation about the input. In terms of like, so that is fine actually, and even though, as I said, the number like error threshold is kind of like bound with the number of PEs. It is not 100% correct because you can circulate data and utilize your number of like the amount of PEs for multiple error iterations, like the inner loop iterations. So it's not 100% or yes, we designed that way, so we don't have to circulate data, but that's always an option. If you want to exceed that error threshold, for example, that's an option. So in the ASIC design, yes, the number of PEs, those kind of things are kind of not very easy to change. But in the FPGA world, it's very easy to tweak with that numbers. And that's what we did in BitMac work anyway. So we tried all those different configurations, and it was just tweaking some configuration file, and that's it. So it was easy in that sense. But yeah. One thing which, one thing which might be a bit more challenging is the number of bits that you process as part of one single PE, because it affects the TB SRAM size, for example, in a very like direct way. So, and as I said, TB SRAMs is one challenge because it's a huge amount of memory you need. So that's one challenge I can see. But other than that, all the other parameters can be easily tweaked.
Great to know that. Thanks.
Yeah. Any other question? Okay, Then let me conclude my talk with like throwing some future work ideas I have. So we can either discuss about them or people can think about them offline. So up to you. So one of them is, I'm not sure if you noticed, but as part of SIGGRAPH, we are just naively doing some filtering, which we cannot even call as pre-alignment filtering. So that because of that, we have a lot of seeds that are being sent to BitAlign, where we rely on the high performance BitAlign module we have. But we don't have a mechanism, like a full mechanism in between MinSeed and BitAlign that performs real pre-alignment filtering. So that's one future work, extension work. And I'm not aware of, like we have in read mapping world, a lot of those kind of separate step like deep dives actually. So I'm not aware of any like pre-alignment filter crafted for sequence to graph mapping, for example. There are some approaches being done as part of the mappers, like sequence to graph mappers, like VG Graph Aligner, but there's no like good amount of comprehensive work focusing on that, like we have in read mapping. So it might be a good research direction, maybe. The second one is GNASM focused actually, and I talked about it. Right now, we provide partial support for different scoring schemes like affine gap model in GNASM. But I believe there must be some ways, mostly algorithmically, to change the underlying algorithm and have a direct support for affine gap model by changing the algorithm, maybe rewriting it from scratch, but keeping the benefits of our new GNASM like algorithm or design and building on top of that with some algorithmic changes, maybe significant of them, but the principles should remain the same, and coming up with full support for those kind of complex scoring schemes might be another future work actually, which I'm very interested in, but yeah, I couldn't do it basically. So the other one is, I covered the read mapping pipeline. We touched some steps of it, but we never claim that we are providing end-to-end support or end-to-end acceleration of the full mapping pipeline, starting from indexing till the very end, like output generation, like cigar string generation or whatever. That's a very comprehensive work, and you should not think of it as just a hardware acceleration work. GPUs are lacking such pipelines as well. For example, especially for long reads, that's there should be more efforts on putting everything in a single architecture so that we will be hiding that data movement problem as much as we can. So that's another future work I can think of, and this is part of my dissertation as well. I gave more like details about it. And the last one is, which Nur asked when I was explaining my first work. So assembly with long reads is one of the main contributors to the end-to-end execution time of the pipeline, the Genome Assembly pipeline, for example. And I am aware of only one acceleration work for accelerating assembly with short reads. It's not even with long reads. And it is not covering all the internal steps of the assembly step either. So, but assembly is another graph-based approach, different than what we have in SIGGRAPH, but it is an important problem for sure. And there is no accelerator work for long reads. And again, my claim for GPUs might be valid here. I'm not 100% sure. So I don't want to say that, but there isn't as much effort as we are seeing in mapping domain that we are seeing in the assembly actually. So it is lacking. So that's another future work, immediate work, whatever you call it, I can think of actually. So that I can suggest to like fresh PhD students or people who are interested in this domain, mostly to our group, of course, but yeah, to anyone. And yeah, this is it. Thank you so much for joining the talk. And I'm happy to answer any other questions if you have.
You must be tired now after 3 hours of continuous.
Is it already 3 hours? Oh yeah.
3 hours plus.
Yeah.
It was a great talk. Thank you so much, Damla.
Yeah, thank you so much for attending for 3+ hours.
So since you completed your PhD and you left our group, of course, we will keep in touch and collaborate for other projects. But I would like to ask for your advice for the upcoming students, if they want to work on this area. What are the things that they might want to focus on as hot topics?
Yeah, so I list some of the related ones of my work here, but I think which I can see from our group as well. I highlighted it in multiple places, but variant calling pipeline is a very complex but important pipeline for sure. It's a standalone pipeline by itself, but it's very critical because read mapping without variant calling doesn't make any sense, for example. We need it. So one, like, and as I said, read mapping is a crowded area right now, and we are shifting the bottlenecks from read mapping to other steps of the pipeline. So I can think of variant calling, for example. We can or future PhD students, our group, Safari, whoever would be interested in, I think should look at that pipeline, one thing. And we touch some of the other ongoing or new research directions as well. For example, like looking at raw data, skipping some steps, or eliminating some computation and doing computation on the raw data itself. That's an interesting area as well. But when I think about more hot topics, more larger topics, I think one thing I can come up with is variant calling, actually. I would suggest that beside what I have already covered here. Yeah.
Thank you so much. Yeah, I agree. I think the things you mentioned also on the slide here, very promising projects.
Yeah.
Actually, each of these points can be several projects, not one project.
Exactly, for sure. It can be some of them can be standalone PhD dissertations as well, like acceleration assembly, for example. That can be a standalone thesis. So yeah, some of them are huge, some of them are more extension projects. Yeah. And regarding your question, I think I can make this comment as well. Yes, as part of my PhD and our group, we mostly or all the time looking at sequencing, for example, which I will touch my new job, current job, where Bionano Genomics is not a sequencing technology for us. They are not proposing a sequencing technology. Yes, we are doing genome data analysis, but it is a different platform, different technology, which is called optical genome mapping, or OGM. Again, the goal is the same, detecting structural variations, but in a different way and in a different scale, not in the base level, but in more as I said, map level, more overview level. So that's another upcoming, I could say, that might be a research direction, because a lot of computation involved in critical computation involved in that sense, in that domain as well. So it might be worth checking too. Yeah, as I said at the beginning, I might be biased, but I cannot be biased because my PhD is on sequencing, technologies and analysis of that. So this is just another way of doing it. So they are relevant. The end goal is the same for all, but the computation is different. So it might be worth checking actually. Yeah.
it is always very good to be independent of the technology because technology keeps changing.
Yeah. And it's totally different. As I said, you are not looking at base level, you are looking at in a larger level. So like an overview, or I'm not sure if this correct term of bird view, maybe. So yeah, so it is different. The technology is completely different, but it might be like worth checking, as I said, because as you can imagine, I believe in the technology and it's an emerging domain. So yeah, that's another thing I can think of.
All right, so do we have questions from, I think on YouTube, we don't have any more questions. I would like to really thank the people still watching us. I don't know if they're watching us from Europe or from US. If this is from Europe, I really appreciate it. It's a weekend over there.
It is, yeah.
Very late.
But we had 1 extra hour today, so maybe people are more energetic because of that. Maybe, I don't know.
Okay, but then let's stop.
Dinner, I would go for it.
Yeah. If there are any other questions, I'm happy to answer.
Yeah, I try my best to be in the shoe of whoever watching us. I try to ask whatever question may come to mind. Please feel free to ask more if you still have some gaps. I think Damla already posted her email, contact details. So whoever have any questions, anything to ask, you can contact Damla directly.
Yeah, please do so. Yeah. I would be happy to answer any other remaining questions offline. Yeah.
All right, it was great to have you, Damla. Thank you so much for teaching us new stuff,
thank you.
Keeping with us for 3 plus 3:30 hours, 30 minutes. So of course, we are happy to have you again.
Same here.
Take care of yourself and we'll be in touch.
Yeah, we will. Thank you so much.
Thank you so much.
Great to see you all. Bye-bye.
Thank you, Damla.